Buenos días a todos. Good morning, everyone, and welcome to this 7th edition of the Young Researchers Symposium of the Rocasolano Institute of the CSIC. We are very happy to be here with you today. So we thank you, the audience, warmly. We have to thank, of course, first of all, the speakers, all those young researchers who are here ready to present their work, their objectives, the way they do things, their methodology, what they want to find out. I would also like to thank the chairs, people who are happy to present the speakers and introduce the discussion and generate scientific interchange. Of course, I have to thank Beatriz González because she's been essential in defining the scientific structure of this meeting. And Esther Rebollar and Antonio Diaz, who have been helping a lot with all the technical issues concerning this meeting, the scientific meeting. For the audience, you can follow this on YouTube, and we encourage you to be very active with the questions. Don't let your curiosities stay with you. Please comment on anything that comes to mind. And a lot of contributions have been received this year. So we have decided to allow everybody, so that the talks will have to be short, but I'm sure that has also advantages, and the speakers will have made an effort to concentrate on the most important bits of their work. I'd like to thank especially people coming from the Unidades Asociadas, associated units, because this is a very good occasion to learn about the work that is carried out in them. And well, I have no more comments. So I think we can start the symposium itself, where the first session will be chaired by Nuria Benavent. So the floor is yours, Nuria.
Thank you, Rebecca. Good morning, everyone. My name is Nuria Benavent. I belong to the Department of Atmospheric Chemistry and Climate, and I will be the moderator of this session 1. We will begin with gas phase reactions. So let's get started with the talk of Javier Carmona García. Whenever you want, Javier.
Hi, everyone. I will share my screen now. Okay, so in principle, you should see it quite all right. Right? Yeah. Okay, so hi, everyone. My name is Javier Carmona García. I'm a PhD student in theoretical chemistry and computational modeling. My PhD thesis revolved around the study of the photochemical properties of atmospheric mercury compounds and how their light-induced chemistry impacts the whole atmospheric cycle of this metal. So for all of you that don't know, mercury is a strong neurotoxin that remains in the atmosphere for long enough to be transported far away from its emission sources. This system can be found in the atmosphere in 3 main forms: elemental mercury and monovalent and divalent mercury derivatives. And therefore, understanding the redox mechanisms that governs the conversion among all these compounds is mandatory to evaluate the fate of this contaminant in our planet. Regarding the oxidation, the main oxidation pathway is considered to be initiated by photochemically produced bromine, but the reduction of the derived oxidized species back to elemental mercury is not well understood, and the photochemical route, and that is the reduction induced by sunlight absorption, is not even considered in conventional atmospheric models. Because of that, we are interested in studying the photochemical properties and photodissociation channels of major oxidized mercury compounds in order to evaluate the impact of this photochemical reactivity in the cycle of this neurotoxin. For that, first of all, we compute theoretical ultraviolet and visible absorption spectra using multiconfiguration and quantum chemistry. And with that, we can obtain band shapes that are in good agreement with experimental data. Here you have an example for a nitrogen derivative of mercury. And with the computed cross sections, we can obtain photolysis rates, which are just first-order kinetic constants that tells us how efficient this light absorption is for these compounds. In the case of mercury compounds in the atmosphere, they strongly absorb ultraviolet and visible light, and therefore that absorption is efficient. However, we don't know if that efficient absorption will end up in a photolytic reaction or a photoreduction, as I said in the introduction. So for that, we can use molecular dynamics. We can simulate using molecular dynamics and multi-configurational quantum chemistry the evolution of our system when it's excited to an excited state, and that excitation is induced by sunlight absorption. And by studying the evolution of bond lengths during the simulation time, we can determine the different photoproducts that we obtain from that efficient light absorption. Here you have, for example, the quantitative photoproducts ratio for this nitrogen derivative after light absorption. And as you can see, we obtain two different kinds of photoproducts with a specific ratio between them. Combining this photoproducts ratio, the photolysis rates, and other thermal reactivity, we can construct a kinetic mechanism that tells us the conversion among all atmospheric mercury compounds. And by analyzing the results that we obtain, we observe that light absorption by major oxidized mercury compounds ends up in an efficient photoreduction that ends up directly or indirectly to elemental mercury, which means that these processes can be the dominant reduction of atmospheric mercury. The final step is introducing that photochemistry in a global chemical transport model to compare the model results with atmospheric mercury observations and check the impact of this photochemistry in the whole cycle of this metal. And for example, as you can see here, there are two different model simulations, one that does not include any photochemistry, and the run 2, in which we include all these photochemical reactions that I told you in the slide before. And here is the concentration, the atmospheric concentration of elemental mercury, and here you have a scatter plot of model to measurement elemental mercury concentrations. And when we include all this photochemistry, here the dots are atmospheric observations, and in red is the model results. We are overestimating the amount of elemental mercury by 99%, which end up giving a long and unrealistically long mercury lifetime, and give us the sensation that there must be some oxidation that we are missing right now, that current atmospheric models does not take into account, and therefore we should try to find this oxidation pathway that is missing and comply the vision of the model results and the observations. And I think that's all. Thank you all for your attention. And in the question sessions, I will answer any questions that you will have. Thank you so much.
Thank you very much, Javier. Nice presentation. Now is the turn of Javier Cachón Ortiz. We are ready to listen to you.
Can you hear me?
Yes,
Hello?
I can hear you. Yeah.
I'm going to share the screen. Okay, give me a second. There. Can you see the screen?
Yes, we can see it.
Okay, so well, I'm going to present myself. I'm Javier. I'm a first-year PhD student at the Chemical Physics Department at the Complutense University. And I was going to talk about the photodissociation of these kind of molecules, the alkyl nitrites, but really we don't have that much time, so I'm going to give a more general overview of the whole thing. So as I said, we are studying photodissociation, which is nothing more than breaking the molecular bonds, the atomic bonds within a molecule with the absorption of photons. Therefore, our objective is to study the different ratios of the different dissociation channels, the different ways the molecule can break apart. In this diagram, you can see that with a water molecule, where once a photon is absorbed, it can break in different ways, depending on quantum effects like quantum tunneling and the selection rules. So the way to do this, first we have to break the molecule, and we produce something we call the Newton sphere. which is nothing more than the fragments, once the molecule is broken, expanding outwards. A good analogy to this would be like a regular explosion, where the fragments will go outward, and depending on their mass, they're going to expand at different velocities. So once we do that, we select the fragment we want to study, and we apply the resonance enhanced multiphoton ionization, REMP, which is basically a multi-particle event where several photons and several virtual states can be used to excite the fragment and ionize it. The reason to use this and not a regular absorption is because this allows us to study the rotational structure of the molecule, of the fragment. So once we do this, we have a electrically charged fragment. Since we took an electron away, we can apply two different methods: time of flight mass spectrometry, which will allow us to select the different fragments if there were multiple ones. You can see that in the image on the right, down on the right, where you can see different lines. In this case, you can only see one because these are preliminary studies, and we only study the NO fragment, and we couldn't only see that. But there could be more, like oxygen or other things. And also the most important part is the DC slicing. This allows us to obtain images of these fragments. The way it works is once we obtain the Newton sphere, we allow it to expand. And once it just before it arrives to the detector, we have a gate which changes the gain of the detector, and we only detect the central part of the sphere, ignoring the sides. We do this because there is where the information we want is. So without getting into much detail, that's basically the whole experimental technique. In terms of setup, we work with different lasers. We have two neodymium jack lasers, which are capable of emitting in 3 different wavelengths, the fundamental one and 2 harmonics. Since we want to tune these wavelengths in terms of which molecule are we studying or the rotational levels we want to see, we have these neodymium jacks go into dye lasers, where the dyes allow us to go into basically within a range of the spectrum. We can go to whichever wavelength we want. Also, we need a vacuum chamber where the whole event happened. You can see that here, where it's divided in 3 parts. First, we have the expansion chamber, where the gas is introduced in a way that we produce a beam of pulsed molecules. We do this because it's necessary to actually see anything, so we can actually synchronize the rates, the pulse rates of the lasers, and we use this valve, which is basically an electromagnet, which opens and closes, allowing the molecules to produce, allowing the molecules to be introduced as pulses. We let them expand in the expansion chamber, and then they arrive to the ionization chamber, where both lasers, the pump, which is the one that breaks the molecule, and the probe, which is the one that uses the REMP technique to ionize the fragments, go into the same point in space. And these Newton spheres, the fragments, are then allowed to expand a little bit more through the use of electrostatic fields to reach the detector. This is the final part of the experiment, where they reach a detector where, through cascade effect using phosphorus, we produce light. This is then captured with a CCD camera. So once we have that, now we can get the images. This is the kind of results we can normally get in these experiments, where you can see clearly the rings, which are the compactness expanded. Here, I didn't want to get too much into the results, since these are preliminary results and there is not much time. But basically, once we have the rings, we normally apply some analysis to study the energetic values in different to study the energy at different places where we can see the different channels. It's normally we integrate radially, or do some, like you can see in this image, and compare them to see which channels are populated, basically. And that's basically it. Thank you.
Thank you, Javier. Excellent talk. The next speaker is Pedro Recio Ibáñez. You can start your presentation.
Okay, let me. I need to wait for Javier to stop sharing.
Yeah, give me a second.
Okay, I hope you're seeing everything. So,
Yes.
Hello, I'm Pedro Recio, and I'm a postdoc student in the chemical physical department of the University Complutense of Madrid, and I'm going to talk about the photodissociation of methylene. But before we see some results, let me talk about the basics of these techniques. Even my coworker Javier Cachón already explained to you. There are basically two main types of photodissociation process. The most straightforward, the direct photodissociation, happens when a generic molecule in the ground state is excited early to a dissociated excited state, from where the molecule, when the fragment will be formed. This process can also take place in a single state if the energy of one photon or several of them are high enough. The other type, the indirect photodissociation, happens when the molecule is excited to a bonded excited state, where if this state is coupled with a dissociative state, the dissociation will occur via an adiabatic coupling between the states. If they are not, through a non-reactive internal conversion to a ground state from where the dissociation will happen, or if the bond state is a quasi-bond state. The dissociation will happen via the redistribution of the between the intramolecular energy or through a tunneling effect. In any case, when the molecules are hit by the laser, the laser beam, they are scattered. The products are scattered in all directions of the space, forming the neutron sphere, which is centered in the center of mass of the molecule. The generated photofragment, as Javier said, they are rationally ionized by the absorption of one or multiple photons using the RMP technique. This technique presents high spectral resolution and to detect the different quantum states in which the molecule is generated. In this case, we use the velocity map imaging technique to detect the Newton sphere. In this technique, for this technique, the Newton sphere is crashed onto the plane of the detector, forming a two-dimensional distribution. However, in contrast to the slicing, this technique requires a mathematical distribution to reconstruct the 3D distribution here to obtain all the necessary information. Since the ejection of the fragment not necessarily the same in all directions, we can determine the anisotropy distribution. That for polyatomic molecules is defined by this equation, where depending on the value of beta, the distribution can be parallel with intensity in the pole of the rings, perpendicular with intensity around the equatorial area of the ring, or isotropic with intensity along the horn ring and defined by the sphere. The experimental setup is a nanosecond laser system, and since Javier already explained to you, I'm not going to enter in these details. But since the detector is sensitive to the position, it requires a calibration to establish a relationship between the pixel of the CCD camera and its corresponding velocity. For that, we need to perform a velocity mapping experiment or a slicing experiment, depending what we are going to use, of a well-known molecule. In this case, we use methyl iodide, since with dissociation can follow these two pathways. Since the experiment was performed with a single laser at this wavelength, we were only able to detect the first one. We can calculate the energy of the fragment by means of this equation, where all the data is already known, and the calibration constant is calculated by this other equation. Notice that the calibration, the constant is valid for identical front-end distance of T1 focusing condition and repellent voltage. An appealing system that has received a considerable interest in the last decade is methylamine, and particularly for photodissociation following the absorption of the first UV band. Mainly because it's the simplest case of ammonia, but also because it can be observed in the interstellar medium, which makes it very important in the astrochemistry and astrobiology areas, since it's also a precursor of amino acids and a majority of heterocyclic molecules. The absorption spectra and electronic state of ammonia and methylamine share several important characteristics. However, the photochemistry of methylamine is far richer. In fact, 5 dissociation pathways have been experimentally identified. And for all of this, we have centered our attention in the first two, the H elimination channels. We have measured the energy, the dissociation of methylamine around the end of the first absorption band in this part. These are the images for all of each of the excitation wavelength and its corresponding energy distribution. You can observe that the energy distribution is divided into 3 different constant functions, which each of them correlate to a different dissociation pathway to the formation of the atom fragment, yes. The peak in the middle, the blue line, is associated to a direct photodissociation in the excitated state, which is coupled to the ground state via conical intersection, forming finally the methylamine radical in the ground state surface. The slower peak, the pink line, is associated, could be the molecule failed to pass the conical intersection in the first attempt and tried afterwards, making the transfer in that of more attempts. This will indicate that the molecule obtained more of the internal energy, which will indicate a lower fragment. The faster peak, the
I'm sorry, Pedro, but you should finish your talk as soon as possible, sorry. Please.
Yeah. The last one is associated to the formation of methylamine radical in the excitated state and has been observed before. So that's all. Thank you. I'm sorry for the delay. So.
Thank you, Pedro. It has been quite interesting. So now we have 10 minutes to discussions. So please, if someone has a question that would like to ask, you can do it using the YouTube chat and I will transfer it to the speakers.
I would like to ask something, if I may, Nuria?
Yes, Rebecca.
Well, in fact, I hope, yeah, it's really a question for the 3 speakers of this session, no? Because I see some related issues. Because Javier mentioned that there are things about the light-induced reactions of mercury compounds, no, that are not completely understood. And I wonder whether the techniques described by the Complutense people would be useful to understand those, or because the Complutense work is mainly like single molecule work, right? Like it assumes that the molecules are isolated, and maybe your situation is different, so it cannot be treated, no, with those methods. It's just for all 3 of you. What you think about that?
Yeah, I was going to write in the chat, like asking general questions to them, because when I was reading the abstract, I noticed, I was like, oh, they are doing something that is something that we sometimes miss, because I think in our case, one of the problems is that for some of these systems, we are predicting the absorption spectrum and then the photodissociation channels, but there are no experimental data for some of them in that will that said that maybe the absorption in this specific absorption band leads up to a photodissociation of this kind, and it's exactly what you are predicting with your experimental setup. And I wanted to ask you both what kind of polyatomic molecules are you keen to study in the future, or what limitations you may have in terms of, I don't know, of size or specific types of atoms. I don't know, just
in size, I don't think that, I mean, as long as we can make a molecular beam of it, I don't think that is a problem of size. It's more a problem of that there is a way to detect this fragment that we could want to detect. For example, the CS3 is it has a very well-known REMP, but there is another fragment like the A2, for example, that it also has a REMP, but it's much difficult to obtain, or other type of fragment that if we could find a way to detect this fragment, there is no problem to do it. So.
Okay, so it's more about the fragment itself.
It's a way. It's more about finding a specific wavelength for this fragment in which is has a resonant absorption. So we are able to select this fragment.
Okay. Okay.
But in time of sites, I don't think that we.
Yeah, no, I was wondering, like, I don't know if which time. For example, I was thinking as well in volatile organic compounds, for example, that sometimes, yeah, in the atmosphere are a lot of photodissociations for this type of system. I mean, and I think the fragments in those are pretty, like, are just.
But it's I mean, obviously the difficult here is more with the site is the difficult remains in making the beam, because if the site is very big, the molecule is probably gonna make clusters, or it's easier to form. Because we want to, we try to make the beams with this pull ring that only a single molecule is ionized when we hit with the laser. If the molecule is very big, we have some problem with it's probably gonna make some cluster, and when we ionize it, it's not exactly that molecule that we are ionizing, you know, a cluster at the end.
Oh, okay.
I mean, it's also interesting. It's another stuff that we can study, but it's not what we want in that moment.
Yeah. Okay, I understand. Okay, thank you.
Thank you. Any questions? No? Well, I have a question to Javier Carmona.
Okay.
You mentioned that you have implemented your investigation on gas phase. Are you planning to do so for heterogeneous phase at some point in the future?
Yeah, we are at the moment. We've just developed a computational strategy first to obtain absorption spectra in condensed phases and in the gas phase water interface. We have a methodology to predict the absorption spectra in those interfaces, and I think that would be the first step, and move to more complex phases like ice, for example, and the absorption spectra in ice. The problem in that is to exactly know how these systems are absorbed in the surface, and a next step in a more like in a longer distance, I guess, is to do these photodissociations in condensed phases, which is a little bit more problematic. But yeah,
okay.
We are on it.
Thank you very much.
You're welcome.
Any question? Okay, so Pedro?
Yes, tell me.
Have you got any specific question or problem where you would like to have feedback from the audience?
No, that I can think right now, sorry.
Okay, so more questions? Okay, so Javier Cachón, I have a question for you.
Yeah.
Yeah. In your presentation, you mentioned two techniques, the imaging technique and the DC slicing. However, you say that you decided to use the later one. Could you explain us a little bit more why you use this technique?
Imaging and slicing. Slicing is an imaging technique. What I meant to say, I probably said something wrong, is that we use an imaging technique, which is slicing, at the same time as time of flight spectrometry. Time of flight to detect, to differentiate between the different co-fragments, and slicing to obtain the images of those co-fragments.
Okay, thank you. Any questions in the YouTube chat? We don't have questions. Okay, so we can move to the second part. So now we are going to continue with 3 advanced materials presentations. Jairo Obando is going to start this part. Welcome, Jairo, at your convenience.
Hello, can you hear me? Yeah.
Yes. Yeah.
Okay, thank you. Hello to everyone. My name is Jairo Obando. I'm a PhD student at the University Complutense de Madrid and the University de Paris-Saclay. In this talk, I'm going to tell you about one of the research lines that we are working together. This has to do with molybdenum disulfide and the modification of its electrical properties when we combine it with an alkaline material like lithium. Well, first of all, why it is interesting to investigate this system? Currently, most of the research is directed towards the efficiency use of renewable energy sources. The efforts, as we can see, that are mainly concentrated in the photovoltaic industry. And however, once we manage to collect all this energy from the environment, if we don't use it immediately, what we can do with this remaining energy? Storing in batteries seems like the most logical option, but again, if we want to be environmental friendly with the earth, what should this batteries made of? One solution is to use transition metal dichalcogenides such as molybdenum disulfides, which is an abundant material in the Earth's crust and whose crystalline structure is ideal for storing energy. This crystalline structure is a van der Waals type crystal, which consists on stacking two-dimensional layers of material strongly bound in the plane, as you can see here, and weakly bound along the stacking direction. This weak binding, in turn, allows to have a lot of interlayer space, which is enough to accommodate charge-donating elements such as lithium or sodium. And it is this removal or intercalation of these elements that is the basis of the operation of charge and discharge cycles in a battery. In here, let me get the laser. In here, you can see 2 cartoons on how we experimentally do the extraction or the intercalation process. For extraction, depending on the amount of material we want to remove, we need to choose an anneal at a defined temperature, or to accompany this annealing with sputtering of neon or argon atoms, depending on the cross-section that we need. And for intercalation, what we do is an evaporation with a commercial solution that allows to evaporate from lithium to cesium alkaline material. What is important is with these 2 techniques, we have full control of the amount of the extraction or intercalation that we want to perform. And also it's important because as these techniques happens in the ultra-high vacuum environment, allows to measure directly the changes and couple these changes to the inclusion or extraction of alkaline materials. To observe the changes in the electronic properties, we use angle resolved photoemission spectroscopy, which allows us to reconstruct the band structure of the material and to observe subtle changes in the valence band. Here you can see a cartoon of the experimental technique, of the ARPS technique. Knowing in detail this region, the valence band region, as well as the evolution of the Fermi level is crucial, because this is how we can quantify the electronic interactions of the material with respect to an external agent. Conceptually, the experimental that we did past winter on synchrotron soleil is quite simple. We just exfoliate a basally oriented sample of disulfide, single crystal, and we measure constant energy surfaces, as you can see here, that help us to know the main crystalline symmetry directions. For example, for instance, gamma K. And on the other hand, we also measure the energy of some internal levels like sulfide, molybdenum, and lithium that give us clues on how the chemical environment of the material is evolving. Then after we repeat the same measurements, but when we after evaporating different amounts of lithium, and what you can see here in this part of the slide is how the valence band evolves as we increase the evaporation time, and therefore the amount of lithium that we're putting on the system. Depending on the temperature of the substrate and the flux of lithium evaporation, we can get into two different situations. One in which the disulfide molybdenum structure is able to diffuse all the lithium homogeneously into the bulk. This is to intercalate it, and another one in which part of the lithium diffused, but another remains stuck at the surface. In our case, we're in the first situation in which lithium is homogeneously intercalated in the structure, and an electron transfer process occurs. This is a lithium just yielding electrons to the disulfide, to the disulfide, yeah, in which the crystalline structure is not modified. Not modified. This is why we can observe the same shifted bands, and even when the Fermi level is has been shifted enough, we're able to resolve the unoccupied band. So I encourage you to ask me in the question sessions what is expected to happen in the second situation that we are not seeing in our case. This is everything. I hope you enjoyed the slides. Thank you.
Thank you very much, Jairo. Nice talk. So the next speaker is Javier Prada Rodrigo. Whenever you want, Javier.
Okay. Can you hear me?
Yes, we can hear you.
Okay. So you should see it all right, I think. Okay, so I'm Javier from the University of Salamanca. I'm a third-year PhD student, and my thesis is co-directed by Esther Rebollar from this institute, and I'd like to talk about laser surface structuring of polymers and nanocomposites. Polymers are interesting because of their versatility and ease of processing, which results in a good quality-price ratio. To improve on these characteristics, two well-known strategies are the creation of nanocomposites and surface nanostructuring. A nanocomposite is created by introducing a small percentage of a nanomaterial in a polymer matrix. Some of their applications are as catalysts or in solar cells and sensors. On the other hand, surface nanostructuring means modifying the topography of a surface in order to change its properties. It has applications in solar cells, organic LEDs, and wearable sensors. This surface nanostructuring technique is usually achieved by lithography techniques, which are well-established techniques with high reproducibility and reliability, but need wide rooms and a controlled atmosphere. I study as an alternative a laser surfaces nanostructuring method called laser-induced periodic surface structures, or LIPS for short. And their main advantage is that all their low equipment requirements, since all you need to do is irradiate your sample with a laser in an adequate energy range to get these linear structures with a period related to the wavelength of the laser and the irradiation angle. My work can be divided into major lines, a more fundamental line where I try to understand the formation mechanism of LIPS, and a more applied line where the interest is in the property changes brought by LIPS and their possible applications. Regarding the fundamental line, we made an experiment where we studied the formation of LIPS in thin films of a polymer deposited on different substrates. We found that LIPS only form for the silicon and glass substrate, but they wouldn't form for the polymer substrate or the steel substrate. To check what influence the thickness of the thin film and the roughness of the substrate had in this phenomena, we simulated the electric field via finite element methods, finding that indeed the roughness of the substrate and the thickness of the thin film can have strong influence on the formation of LIPS and their characteristics. In particular, when the roughness is too high, it can break this periodicity, which would inhibit LIPS formation. So our current hypothesis is that for all metallic substrates, LIPS would form, but since it's too rough, they are inhibited, which was reinforced by another experiment we do where we try to generate LIPS in the same polymer over two gold substrates, one with high roughness where LIPS didn't form, and one with low roughness where LIPS did form. Regarding the semiconductor substrate, it would act like a metal while the irradiation takes place due to the excitation of electrons induced by the laser. And since the roughness is low, we see LIPS forming. However, for the dielectric substrates, we see lips in the glass substrate, but not in the polymeric substrate. And we are not sure why yet, but we are performing a new experiment to discover why. In the applied line, we irradiated a polymer and a nanocomposite based on it with laser pulses and characterized the changes in their topography with atomic force microscopy and their nanomechanical properties with peak force quantitative nanomechanical mapping, which is a technique based in atomic force microscopy. And we used the sessile drop technique to measure the contact angles of different liquids before and after irradiation. And from it, we obtained the wettability of the sample and the surface energy. We find that the same changes for the polymer and the nanocomposite were induced by irradiation, which were that in the topography, we found that the structures have a period of 350 nanometers and form parallel to polarization. From the nanomechanical measurements, we find the Young modulus is preserved, but there is a decrease in adhesion. And from the contact angle measurements, we find that there is a hydrophilicity increase, but the total surface energy remains constant. Moreover, we have started experiments to determine the feasibility of using lips as optical resonators. And in order to do that, we need lips of a specific period. So if you remember the formula from the beginning, we need to irradiate at an angle. We have done some tests, but we're still trying to find the correct energy range. We're also working in the creation of surfaces with different microstructure areas for lab-on-a-chip systems. And for this application, we use another laser processing technique called laser interference lithography, where we use the interference of two laser beams and the period of the structures is a function of the angle between the beams. We have also yet to find the correct energy range for it. Thank you all, and that's the end of my presentation.
Thanks, Javier. Very good presentation. So, and to end the first session, it is now the turn of Marina Gabriela Martinez. You can start your presentation.
Okay, thank you. Just a moment. I can share. Javier.
Oh, yeah, sorry.
Okay. Okay. Good morning, everyone. As Nuria said, I'm Marina Martinez from the group of Laser Nanostructure and Material Processing. I did my studies in conservation and restoration in cultural heritage, and currently I'm finished my high intro scholarship. I'm going to do my presentation on characterization of medieval-like glass alteration layer by laser spectroscopy and nonlinear optical microscopy. Historical and archaeological glass is a cultural heritage that needs particular care when studying it due to its inherent fragility, and careful manipulation of the glass can result in the loss of its integrity. Studying glass provides us with good information about the manufacturing process via the study of the glass composition. The environment that the glass was exposed to can also be noted by analyzing the degradation layers. Factors that are related to the degradation of the laser can be extrinsic or intrinsic. These factors are relevant regarding the physical and chemical integrity of the object. One of the alterations that we usually find is the multi-layer structure in the body of the glass. Studying the characteristic and degradation process of the glass can help us to better design of conservation strategies. In particular, in this investigation, our objective is to apply a non-invasive techniques to study heritage glasses. In order to determine the effectiveness of the laser techniques, we perform a multi-analytical approach to the sample through the comparison of two sets of techniques, the traditional and invasive techniques and non-invasive one. The samples are a group of medieval-like model glass under various environmental and atmospheric conditions in order to generate alteration layers of different characteristics. To study the multi-layer structure and composition, traditional techniques include the use of cross-section to gain access to the inner part of the glass body. The surface and stratigraphy of the glass samples was studied by optical microscope, and their chemical composition was determined by scanning electron microscopy energy dispersed X-ray spectroscopy, SMEDS. These techniques are destructive. Techniques of nonlinear microscopy were recently introduced into the study of cultural heritage, particularly in his modality of multiphoton excitation fluorescence. Based on excitation with ultra-fast and ultra-short titanium sapphire laser, the excitation of the material take place in the focus where the nonlinear processes are induced. The collection of the multiphoton signal is carried out in the reflection mode through the same objective and then sent to the photomultiplier. These techniques allow for a three-dimensional characterization of the structure and thickness of multi-layer and multi-component substrates. In this graphic, we can see represent the thickness at the full width at half maximum, resulting from a Lorentzian function finding. Also, we observed a good agreement between the multiphoton excitation fluorescence and the optical microscope. It should be noted that the nonlinear optical microscope was developed in our institute, and until we have now, this is the first time that it's using this non-invasive method for study degradation layers of the glass. Also, we use the laser-induced breakdown spectroscopy and laser-induced fluorescence techniques to determine the elemental and molecular composition of the glass. Moving to the results, firstly, in relation to the composition of the glass, apart from providing complete chemical information at molecular level, the obvious benefit of applied laser spectroscopy is indeed its non-destructive character, as well as its capacity of in situ analysis. Secondly, as we already said, concordance is shown when comparing the thickness of various glass alteration layers and as determined by multiphoton excitation fluorescence and optical microscope in cross-section modality. Overall, we can say that there is a good agreement between the invasive and non-invasive methods. Finally, as conclusion, the proposed methodology, which involves the application of photonic laser-based spectroscopy and nonlinear laser microscopy, exemplifies the possibility to carry out a non-destructive composition and structural characterization of heritage glass item without a sample preparation. Thank you very much for your attention, and thanks also for the received fundings. And if you have any question, I will be happy to answer you.
Thank you very much, Marina. Very interesting topic. So, as we have done before, it is now time for some questions and discussions. So feel free to ask whatever you want in the YouTube chat, and I will transfer them to the speakers. We have a question to Jairo from Juan de la Figuera.
Yeah, go on. I can hear it.
What is the minimum temperature to achieve intercalation of the Litio? Also, where does the intercalation take place? Steps, defects?
Well, very difficult question. As far as I know, the intercalation in this material is not just reliable on the temperature. Indeed, also with the flux of the amount of the material that you're evaporating. So it's a combination of two things: temperature and flux. For each to overcome to threshold these two regimes. In my case, I did my experiments, the evaporation at room temperature, because well, we were not completely sure about the flux amount of lithium that we were giving to the sample, because it's somehow complicated to quantify how many lithium are you putting in a sample. So this is why we thought it would be a good idea to use room temperature and use different amounts of lithium, and to see in which one we were intercalating everything, or intercalating just a bit and another part stuck at the surface. But yeah, that's I don't know if that answered the question.
Okay, thank you very much, Jairo. Javier, want to add something?
Yeah, I had a question for Marina, and I'd like to know if the laser techniques allow you to penetrate the whole crystal, or do you only have information of the surface of your crystals?
Okay, we are with a nonlinear optical microscope. What we are is checking the surface, and after are going into introducing into the surface of in this case of the glass, and in this way we can control the thickness of this layer that we are trying to examine.
Thank you very much.
Thank you. Rebecca, do you want to add something?
Yes. Thank you, Nuria. Yes. Well, just one comment, because Jairo wondered whether the question had been answered. So please, Juan, through the YouTube, if there's something more you want to clarify, please let us know through YouTube. And then I'll take the opportunity to ask a question myself for Javier. This is just about the laser-induced structures in surface, which tend to show a certain degree of disorder, no? But for some applications, one would like more order, right? Less disorder. And so it's a combined question, no? In what circumstances have you found less disordered structures? And if one wants to go in that direction, should one work in the laser field or should one work in the material?
Well, both the laser and the material are important for it, but normally if the material that lets you work with it a bit, if you can get into an energy range, into a fluence range, which is a density of energy and number of pulses range, where the structures are very ordered. So you have to kind of optimize those two parameters, and then you'll find well-ordered structures normally.
Mm-hmm. Okay, thank you.
Okay, more questions? Okay, I would like to ask something to Marina.
Yes.
Yeah,
Okay.
I found your topic very interesting. My question is, could you apply the same emerging techniques to any type of material?
Actually, the multiphotonic excitation fluorescence is based on the measurement of the intrinsic fluorescence from the materials, and you just need to have some. Yeah, can you hear me properly? Okay, sorry.
Yes, we can.
The materials that you want to analyze must have presence of the fluorescence emission.
Okay, thank you. Juan de la Figuera say thanks for Jairo again. Whatever you think, the intercalation takes place through the effects or through steps. Do you want to say something, Jairo?
Yeah. Well, when you're using a basal sample of disulfide, this is the 0001. The intercalation takes place through the effects and also through the own structure, because I don't know if may I do it? I don't know if I have time to share my screen again, because it will be graphically better.
Yeah, you can share it. No problem.
Okay, thank you.
Yeah.
Thank you. Up. Okay. Yes. So as I was telling to Juan, the intercalation can happen whether if you're using basal planes or stepped surfaces, because this when you're putting lithium, this helps at very first to form a top layer on the surface, and this layer downs the potential barrier to overcome lithium through their own structure. This has to be with stiffness of the structure, and this is why the intercalation can happen at first with a basal oriented sample. At first through the structure and also through sulfur defects, and when you're using stepped surfaces oriented, the intercalation takes place through the interlayer space.
Thank you, Jairo. Any question?
Okay, Nuria, I think you have your audio off.
Any question? Sorry. We don't have more questions in the chat. Okay, if there is no more questions, I think we can wrap up this first session and Rebecca is gonna say something.
Okay, thank you very, very much, Nuria, and thanks to all the speakers of this session. I think it's been very, very fruitful and very interesting. So we've looked at molecules and we've looked at materials, and we are now going to go biological now to a session that we call biosynthesis and biomolecules, which will be chaired by Inmaculada Pérez Dorado. Inma, it's your turn now.
Thank you very much, Rebecca. Good morning to everyone. My name is Inmaculada Pérez Dorado. I'm from the Department of Crystallography. And yes, as Rebecca said, I'm gonna moderate this second session. And it's my pleasure to get started with the first talk that will be conducted by Isabel Cea Rama. So please, Isabel, when you are ready.
Good morning, everybody. Can you hear me?
Yes.
Yes.
Okay, so I'm going to share my slides. Can you see it? Can you see it?
Yes.
Okay, so good morning, everybody. My name is Isabel. I work in the crystallography department and I'm a PhD student. And I'm going to present today some of my thesis results. So to start with, enzymes are proteins acting as biocatalysts. They enable faster chemical reactions by lowering their activation energy. They present certain advantages versus traditional chemistry, as we all know. And therefore, because of these advantages, the next goal would be their industrial implementation. But this task remains a challenge because it is very time consuming and it requires an economic effort. And there are also certain required features to improve, such as the enzyme substrate scope and their operational stability limitations. So to overcome these issues, there are two approaches: the protein engineering and the searching for new enzymes. So I'm going to focus this speech on the second one. Metagenomic techniques have arisen as powerful tools that enable you to directly isolate environmental samples without the laboratory cultivation step, which is a bottleneck step. Industrial most demanded biocatalysts, as you can see here, are hydrolases, and within this group, the Hester hydrolases. So I'm going to focus this speech on Hester hydrolase 3, which belongs to family 4, and it is included in the superfamily of alpha beta hydrolase 4. As you can see here, it presents two domains: the cap domain, which is allocated on top of the catalytic domain, where you can see the catalytic triade, and we were able to capture a substrate mimicking the tetrahedral intermediate. You can see it here, the serine, the nucleophile, the histidine acting as a base, and the aspartic acid as the acid. There are also 3 glycine residues. They stabilize this tetrahedral intermediate by hydrogen bonding. EH3 is a very promiscuous enzyme, but it is not stereospecific. So to understand the molecular basis behind its promiscuity, it has a very flexible loop located on top of the catalytic triade that can modulate and can adopt certain conformations about binding, substrate binding. We were able to see that it has a very prominent catalytic pocket, which 3 different binding sites: the alcohol binding site, the acyl binding site, and there is also a 3rd channel which triglycerides can be allocated. So at first we can think that stereospecificity and promiscuity are not compatible features, but I can tell you today that in fact they are, The first approach I would like to tell you is the supramolecular engineering. This is a very sophisticated approach. It has two steps. The first one is the immobilization steps. It is developed through the lysins on the surface of the protein. And the second step is to create a protective layer, and by changing the thickness and composition of this protective layer, we are able to gain stereospecificity properties. And the second approach I would like to tell you is the site-directed mutagenesis. So the X-ray crystallography structure allowed us to see that there was an isolate leucine inside the catalytic pocket very close to the substrates, and by mutating this residue to phenylalanine, this residue played a key role in gaining stereospecificity because it enabled the preference for one enantiomer versus the other one. So finally, I would like to thank the whole crystallography department, special mention to Julia Sanz, and also the collaboration with Manuel Ferrer and his group. And I would also like to thank European funding inside the project INMARE and the recently granted project Future Enzyme. So thank you, everybody. It was a pleasure.
Thank you very much, Isabel, for your nice talk. Please don't be shy. Any questions? Oh, sorry. Apologize. No questions now. I have to give. Now I have to introduce the second speaker. My bad. María Ángeles Márquez, please, when you are ready.
Good morning. Can you hear me?
Yes.
Good morning. I am María Ángeles, and I am in the 4th year of my PhD in the Department of Crystallography and Structural Biology. In this talk, I would like to introduce all of you our contributions related with the crystallization of an enzyme implicated in the inositide signaling. Sorry. Inositides are a big family of small molecules sharing an inositol ring, which participates in a lot of cellular processes. There is one subgroup that is called inositol pyrophosphates, which are those with more than 6 phosphates attached. Inositol pyrophosphates are implicated in a lot of cellular processes, such as phosphate homeostasis, apoptosis, or vesicular remodeling. There is a wide variety of proteins that control the equilibrium between the different inositides. One of them is a yeast pyrophosphatase called DP1, which can hydrolyze pyrophosphate bonds of IP7 and IPH to produce IP6. But DP1 is a promiscuous enzyme that also can hydrolyze diadenosine and inorganic polyphosphates, both implicated in phosphate regulation, sensient, and storage. In this talk, in this work, we want to study the inositol pyrophosphates, and we want also to study their implications in salt by obtaining the two-dimensional structure of DDP1. We want to answer questions about this enzyme, as how can its active site allocate 3 substrates that are so different? First, we have studied a DDP1 structure by protein crystallography. We have produced crystals, and later we have measured high resolution X-ray by obtaining diffraction data and to obtain the DDP1 two-dimensional structure. From this data, we have now the structure of DDP1 alone and in complex with its 3 substrates mentioned before: inositol pyrophosphate, diadenosine polyphosphates, and inorganic polyphosphates. If we make a zoom of the active site, we can see that DDP1 uses the same residues to bind the different ligands. This is the key of the promiscuity of DDP1, because it has a lot of positions for phosphates in the active site thanks to a lot of positive charge as arguments that neutralize the negative charge. In the case of inositides, we have found that they can be anchored in the active site in 5 different binding modes because of the inositol ring symmetry. But just 2 of them are productive and locate the pyrophosphate bound in the reactive position. Here you can see a scheme of the inorganic polyphosphate in the active sites, which is the ligand that occupies the majority of the phosphate sites in the structures we have solved. We have produced, designed, expressed, and purified a total of 11 mutants of DDP1, which is there. And we have also crystallized some of them. We have also compared the biochemical properties by thermal stability and activity assays to know better the function of the active site and the implication of each residue in the active site. To conclude, we have solved the structure of the DP1 in presence of the 3 types of ligands and in different binding modes, as in the case of inositides. We have also understand the promiscuity of DDP1, which is highly related with the multiple phosphate positions found in the active site. And we have also identified DDP1 residues that are critical for enzymatic biochemical properties as thermal stability or protein activity. And now we are focused on the generation of a mutant that can discriminate between the different classes of ligands and the developing of DDP1 inhibitors that can be potential candidates to fight with fungal infections. And to I will thank all people involved in this work, especially my department team at the Instituto de Química Física Rocasolano. I want also to appreciate our collaborators and our founders. Thank you for your attention.
Thank you very much to you, Maria Angeles. Please, if you have any questions, I remind you, you can put them on the chat, YouTube. And continuing with the next speaker, that will be Cristina Cabrero. Please, Cristina, when you're ready.
Yeah, thank you, Inmaculada. Hello, everyone. I'm Cristina Cabrero, and I'm a predoctoral researcher at the NMR Nucleic Acids Group in the Rocasolano Institute of Physical Chemistry. Today, I'm going to talk about modified nucleic acids and the role in the stability of the structures where they belong to by means of nuclear magnetic resonance. Starting from the very beginning, the nucleic acids are biopolymers constituted by a sugar moiety, which is a ring of five members in both DNA and RNA, a phosphate group linking the nucleotides and forming the backbone of the strand, and a nitrogenous base that can be a purine or a pyrimidine. The nucleic acids also fall into different types of structures. As we can see here, they are always helices, changing the number of strands involved. Here, we are focused on the canonical double helixes. They are antiparallel, so the 2 strands run in opposite orientations, right-handed, and the hydrogen bonds formed are of the Watson-Crick base pairs type. One of the most interesting applications of the nucleic acids in the biomedical field is the silencing genes-based therapies, such as the antisense ones. In this context, the RNA is considered a therapeutic target. Thus, an antisense oligonucleotide sequence complementary to a certain region of the RNA binds to this messenger RNA, silencing or degrading it. However, these oligonucleotides are so unstable because they are recognized in the cell. That's why it's necessary to include chemical modifications to stabilize the hybrids with the RNA in order to increase the binding affinity and the resistance against ribonucleases. Here, we are going to study modified nucleotides with fluorine modifications in the 2' position of the sugar, such as FANA and FRNA with an only substitution, and the gemcitabine, a well-known drug used in anti-cancer treatments, which is a cytidine analog with fluorine atoms in both 2' and 2" protons. Other sugar analogs are log nucleic acids, in which the ribose is modified with an extra bridge connecting the 2' oxygen and the 4' carbon. The preferential conformation of these oligonucleotides is related to the stability of the duplexes discussed here. combining the pattern of LNA, FANA, and FRNA, and insertion of DFDC in the DNA strand of a DNA-RNA hybrid as a helix model, and a self-complementary DNA duplex as a big helix model. The methodology followed in this work starts with a presearch of the best condition of temperature, pH, or ionic strength in order to acquire the best spectra. Later, we assign almost all the signals present in the two-dimensional spectra in order to extract experimental information about the distance restraints from NOE intensities and angular torsions from G-coupling in Cauchy experiments. Finally, we use all this experimental information to calculate the restraint molecular dynamics using number 18. The preferential in the first structure, we can in the first structure, the thermal enhancement has been seen, whereas FRNA and LNA shows a north conformation and the stabilization related to the base is taken in pairing, in FANA is due to the forming bonds between the fluorine atom and the H proton of the purine, adopting a south conformation. On the other hand, the gemcitabine is an adaptative nucleotide able to adopt different conformation depending on its surroundings. The result is a destabilization effect due to the alteration of the electrical charges of the near nucleotides. Thus, the FDC adopts a north conformation in pure DNA and a south conformation in DNA-RNA hybrid, being opposite to what we used to expect. This south conformation shows similar interactions to the pseudohydrogen bonds described in the previous structure, compensating this way the base pairs distortions. Thanks for your attention.
Thank you very much, Cristina. Beautiful talk. Well, remind you about the questions, please, for the speakers, and I would like to introduce to the next speaker, Elena Jimenez Ortega. Please, when you're ready.
Okay. Can you see the slides? Sorry?
Yes.
Can you? Okay. Good morning. My name is Elena and I study my PhD at the Department of Crystallography. And I would like to talk about some of my thesis results.
Sorry, Elena, just one, sorry about the interruption. We can see your screen, but I think it's not in full screen mode. So we see the current screen and the next one. So if you can, that would be.
This or? I have to change?
At the upper part of the PowerPoint, you should be able to change the view. I think when you go, oh my gosh, cambiar de partida. Oh. You have to several different modes to show your PowerPoint.
It's the same from your perspective, right? You see.
Yes, I can see.
But this is my problem.
Usar, if you go back, please. I think it's at the top of your PowerPoint presentation.
Yes.
Now I cannot see it anymore, sorry, but it says, can you go back?
If you can go to PowerPoint.
Yes.
We might be able to.
Yes, but you can, sorry.
I see your screen, but
yes.
Just
this is, you can see this?
No.
No, you have problems with.
So I think I cannot see it because I have something on the top, but there is at the top.
Yes.
The second option.
Yes.
I think if you click there.
The second option.
Yes. Is that I cannot see what.
No.
Usar presentación con diapositivas.
Yes, but this is my. I have to.
Okay, if it's not easy, we can leave it as. I mean, it's visible. It's just it's smaller.
It's smaller. But maybe I have to change the screen. Sorry. No, I have a problem with this.
It doesn't go full screen.
But I think it's okay.
Yes, it's okay.
A lot better. Yep.
Okay.
That's
you. Sorry, I have to start. Sorry. Good morning, my name is Elena and I study my PhD at the Department of Crystallography, and I would like to talk about some of my thesis results. sorry, because I can see the complete screen here. I have a problem with the presentation, sorry. Because I can hear in my screen, sorry.
We see your full screen fine.
Yeah, yes, but I see the participants in the presentation, sorry. Yes.
Maybe if the other presentation mode was easier, we could still see it. Maybe it's much easier to go back to that one.
This is, but
it's fine for us.
No. Yes. No, it's difficult in this.
So
because
go back
it's
to
so
the previous,
strange.
Go back to the previous show mode.
Yes.
It should be fine.
The previous mode. Okay.
Yes.
Yes, it's okay?
Okay,
yes,
let's
ahead,
stay
no?
there if it's
Okay.
easier for you.
Okay. But I would like to present some of my thesis results. Let me introduce some ideas. Firstly, the microbiota is involved in many essential processes, and there are some strategies to modulate it. But in this presentation, we are focused on prebiotics. The idea is that we can use the raw material, such as a polysaccharide, and it could be enzymatically transformed into a value-added product that is the prebiotics. Well, another important health problem is the increase of the number of lactose intolerant people, and this problem could be solved by the application of one enzyme in order to hydrolyze the lactose molecule. We study the three-dimensional structure of the proteins that can produce the prebiotics and can hydrolyze the lactose molecule with X-ray crystallography and with cryo-EM. There are several kinds of prebiotics from different polysaccharides, and they have different effects on our health. But in this presentation, we are focused on three: the lactose derivate, silane derivate, and chitin derivatives. Firstly, the beta-galactosidase is an enzyme that can hydrolyze this lactose molecule in order can hydrolyze this molecule lactose. And then also it has the ability to create new links using this lactose molecule as a template in order to produce the galactoligosaccharide. Moreover, there are some enzymes that can hydrolyze this complex oligosaccharides in order to produce another prebiotic, the silooligosaccharide and the chitin oligosaccharide. We study the three-dimensional structure of this beta-galactosidase at high resolution by cryo-electron microscopy. As you can see, we solve the map with a lot of details, and we can see the new assembly, the octamer assembly. And this new assembly is the responsible for some of the properties of this protein, that is the thermoresistance and the stability. This new assembly is thanks to a new adhesion-like domain, this C-terminal domain, and we study this C-terminal domain and the interaction in which participate. And the reality is that we can describe the assembly, the octamer assembly, like a tetramer of dimers. With this structural information and with the 2D negative stain resolved by electron microscopy and the and with these assays were essential for design the best immobilization method for the industry application. We study also the structure of this silanase in complex with linear and with branches oligosaccharide. And the most important result is that this enzyme can accommodate the decoration, the oligosaccharide with decoration. And this is thanks to high flexibility in this sugar, in this position. And this property has a lot of interest in the industry because the enzyme can apply with the traditional silanase in order to increase the efficiency in the silane degradation. Yes, and finally, we solve the structure of this chitinase in complex with different ligands, these 3 ligands, and we identified and mutate different key residues in the catalytic center in order to modulate the activity of the enzymes to produce the chitooligosaccharide that are the prebiotics. And the most significant result discovered was the movement of this domain that can simulate the real substrate binding and the product release of the enzyme. And I would like to thank to my group, and I would like to highlight the collaboration with ENIO that was essential for the cryo-EM study and for the glycogens consortium that are formed by several groups. And thank you very much. And sorry for the inconvenience.
Thank you very much, Elena. Very nice work. Well, now remind you that it's time for questions and for the discussion. Okay, I can see a comment from Armando Albert. Congratulations, very nice work. And I have also a comment from Maria Jose Sanchez Barrena.
Okay.
So Maria Jose is making a question, but could you please tell me? I guess it is for Elena. Please, Maria Jose, can you?
I have a problem to disconnect the screen. My screen with the Zoom, so I don't know how to.
I have
because I have this.
I think you need to stop.
Yes. Okay.
Okay.
So
I have a question from Maria Jose that is for Mari. Mari, nice talk. Have you already thought about the ways to inhibit this enzyme with the small compounds that might constitute therapeutic hits?
Yes, well, we have to start with the inhibition of the protein because a lot of our inositide structures eat with inhibition inositides. For example, I have an IPH, this is IPH, red are phosphates, and we have pyruvates pyrophosphates. So we can't use an IPH with a carbon here and here that are not phosphate and cannot be hydrolyzed, and we have the structure with this compound. It's called PCP-IPH, and it could be a potential candidate. We have also the structure with PCP-IP7, PCF, which is a different inositide molecule, which is not a natural substrate. So we have a start with this, but the problem is that the binding of the protein is really similar to the substrates and the product, which is IP6. So we think it's difficult that the inhibition molecule displaces the inositide substrate, but it is we are working on it, and we have also find another molecules. Thinking in diadenosines or polyphosphates modified that can inhibition protein DDP1.
Okay. I have a follow-up question, but I think you may have partially answered from Maria Jose. So given the different, it is for you, Mari, given the different ways to recognize the substrate, the natural substrate, which would be the best strategy, I guess, in order to generate these hits?
Well, I think the best strategy is to find similar molecules that occupy a lot of positive or phosphate sites, because if when the ligand has not enough phosphate sites, we have seen that the binding is worse. So we need a lot of positive sites occupied, but we have to block the hydrolysis part. So I think the best is to change the part next to the catalytic site and try to the rest to be as similar as substrates it is. Yeah, I think it's the best strategy.
Thank you very much. Well, I have another comment on YouTube. Congratulations to all of you from Juan Hermoso. And I have another question from Maria Jose. In this case, it's for Elena. So which were the keys to obtain such good cryo-EM? Pros and cons to do crystallography or cryo-EM with these kind of enzymes.
For example, for the enzyme that we can see in the slide, we try firstly by try to resolve by X-ray crystallography, by due to the dimension of the molecule, because it's too high, it's a lot of residues. The resolution was not at high resolution by X-ray crystallography, and we decided to try with cryo-electron microscopy because the weight of the sign is appropriate for these techniques, because it's a direct technique and not have the boldness step that you have to crystallize the enzyme, because it's too big and it's less stable than to get the crystal in this case. But in other case, you can apply the X-ray crystallography.
Thank you very much. I have a question from, I'm really sorry because there is lots of things in the chat. Rebecca, I think you have spotted that question maybe better than me.
Well, there was I saw that Javier Carmona said that he had a question to Cristina, so maybe he can go live now.
Yeah. Sorry, maybe it got lost in the chat before.
Sorry, thank you.
Cristina, nice talk. Well, I'm sorry I'm asking this because I'm a theoretician and maybe it's too technical or something, but I was wondering regarding the molecular dynamic simulation, if you can tell me a little bit more about if you are using classical molecular dynamics, using force fields or reparameterized force fields for these systems, because I'm only, it was a long time ago that I did some simulations using, I think the largest molecule I used was the calanine. So, and I was wondering if you can give more details on that.
Yeah, what we do is to put the structure in a water box. So then we minimize this solvent in order to accommodate it with the structure. And well, minimization, thermalization at 25 degrees because it's the
standard,
normal. Yeah. And then the calibration. So then we run the dynamics and we use force field and the classical point of view.
Yeah, and for how long? Just
two consecutive runs of 500 picoseconds.
Okay. Yeah. Perfect.
So 1 nanosecond at the end.
Thank you. Thank you so much.
Further questions for this session? Okay, if there is no more questions, I have one for Isabel actually. So Isabel, I'm very curious about, could you tell us a little bit about the industrial applications of the enzymes you are interested on, please?
Yes, so as I presented today to you, EH3 has a very impressive 71 substrate scope. So it could be used in different sectors, such as, for instance, detergents, the paper and the palm industry, but also in the pharmaceutical industry, because as I presented today, it could be gain the stereospecificity, properties. So it could also synthesize some optical pure compounds, which are very demanded building blocks in the pharmaceutical company. So I guess this enzyme has a very wide and broad industrial application in different sectors because of its properties.
Very interesting. Thank you very much. More questions? Well, if there are no more questions, I think I didn't miss any. I have another question for Elena. I think it was the beta galactosidase. I'm curious because you were highlighting in red a domain. I understood that this domain is not conserved in other enzymes of this class.
Yes.
Do you know what is that domain for?
In other enzymes, beta-galactosidase enzymes, like E. coli or Cluberomyces, that is the most studied beta-galactosidase. They are the classical domain, the 5 domains, but this is an extra C-terminal domains that is the responsible for the assembly, because the other proteins have a different assembly and different oligomeric state, like a tetramer, and this is a special, it's an octamer, and this is thanks to the interaction, the extra interaction between the C-terminal domains, and this is the responsible that for the thermoresistance that can resist 19 Celsius degree or more or less.
I see. Thank you. Thank you very much.
You're welcome.
Okay, I think that we have arrived to the end time of this session. So I would like to say thank you to all the speakers for the brilliant talks. And I think that now it's time for a coffee break. And well, Rebecca, I don't know if you have something to add.
No, that's fine for me. Thank you very, very much, Inma. Thank you, Nuria, too. Thanks to the chairs. Thank you to all the speakers. You've been great. You've been clear and concise. You've been really fantastic. I couldn't imagine that we'd be starting the coffee break exactly at the expected time. So thank you all. We will continue in about 10 minutes. Yeah, we will reconvene at 11:40 as scheduled with 2 more sessions. So please stay with us. We will see you in a few minutes. Well, we reconvene now. We now continue with the 7th symposium of young researchers in the Rocasolano. So now we are going to have the 3rd session of the morning, and the chair will be Luciana Luque. Luciana, please.
Yes, here I am.
Hello.
For the introduction. I'm Luciana Luque. I'm a postdoc researcher from Argentina, but now I'm doing a collaboration stage here at the Rocasolano, particularly in the group of statistical mechanics and condensed matter. So now we are starting our first block for the 3rd session, which is called Understanding Solutions. And our first speaker will be Cynthia Pulido Lamas. So Cynthia, whenever you want. We cannot hear you, Cynthia. I don't know if you are.
Okay, I will try now.
Now, yes, yes.
Let's see. Can you see the full screen?
Perfect. Yes.
Yeah, thank you. So, hello everyone. My name is Cynthia Pulido. I'm a 2nd year PhD student at University Complutense. And today I'm going to talk about computational molecular dynamics as a useful tool to understand how different salts impact water properties. So to begin with, I will give small motivation. So why is understanding ice nucleation important? Well, it affects our daily life experience, and it's really important for climate change, for example, also for food companies, since they have to preserve the food in different ways depending on this phenomenon. It's also useful in cryopreservation of human cells, or to avoid formation of clatrates in the pipelines of oil. These are macro scale phenomenon, but to understand it, we have to look at the nanoscale. And these phenomena are usually difficult to observe experimentally because the conditions are difficult to reach, and because it takes time in a really small time and also small space, so in the nanoscale. So usually we studied by means of computational simulations. In this case, we're going to study it by means of molecular dynamics. We are going to focus on the freezing point depression, which is the difference between the melting temperature of water and the melting temperature of water with salt and in different concentrations. Well, I will explain shortly what is molecular dynamics for those who don't know. We begin from an initial configuration, initial position, and then we apply the Newton equations of motion. The computer does these calculations, and then we obtain a trajectory. We obtain different positions, and from it we can calculate the thermodynamical properties of the system, for example. And in our case, our system will look like this. We have a salty aqueous solution, and we will simulate it by with the Madrid 2019 model. We have a part, there is a block of water with some molecules of salt. The studied salts are the ones that we have listed in the screen, and then we have a block of ice. We employ the direct coexistence method. This example is for pure system. So we begin with a solid and a fluid. If we simulate this system above the temperature of coexistence, the system will become fully a fluid. It will melt. But if we are below the coexistent temperature, which is the melting temperature, all the system will solidify. So we do different simulations at different temperatures, and we would be able to estimate this coexistence temperature. But when we have mixtures, what happens, like in the example for lithium chloride, we have also a certain concentration of the fluid. So here we simulated at a temperature of 220 Kelvin, which is below the temperature of coexistence for molality of the solution. So what happens is that the ice will grow, but at some point it stops. We reach equilibrium. Why? Because when it has solidified part of the water, now we have less molecules of water for the same molecules of salt, which rises the concentration. And now at this concentration, the temperature of the simulation is the one of equilibrium, is the melting point. And as we see for mixtures, we only need one simulation. We do not need several, like for pure systems. So here we have an example in the phase diagram of what we did. The blue marks indicate where we simulated a system. This system was the same one, and we run it for 3 different temperatures. And we can see that when we started at a temperature that is really similar to the coexistence ones, the system did not move. The concentration of the solution was the same at the end. And when we started at a temperature that is above of this of coexistence, part of the ice melt. So now we have smaller concentration in the mark above, and in the one below.
Cynthia, you have to finish already.
Okay, in the one below, the temperature is below this of the coexistence. So the concentration got raised, and now we can calculate the temperatures. We did it for different salts, and here are the results. The lines are for experiments. As we can see, the simulations predict quite well what we have seen via experiments, also taking into account that experiments had to be fit because there was a large dispersion of the results that we do not get. And thank you for your attention. This is all.
Thank you so much for your nice talk. So next we have Lucía Fernández-Sedano. Lucía, if you want to share your screen.
Okay. Well, Lucía Fernández-Sedano. I'm an undergraduate student at the Complutense University of Madrid, and this work has been done in collaboration with the University Complutense and with the University of Vigo. And we have studied the supercooled region of water that is below the melting point to determine the density curve of water solutions. Water presents a maximum in density at 4 Celsius degrees, and this anomaly is which makes ice less dense than water, and so it floats on water. This is important in small masses of water in very cold climates, because as the water cools below 4 Celsius degrees, it rises to the surface because it is less dense than warmer water, and it ends up forming a nice layer. And this layer shields the water below it from the exterior, and so it maintains the temperature between 0 and 4 Celsius degrees, and the living organisms can adapt to these conditions. Having an electrolyte force field that faithfully reproduces the ion-ion and water-ion interactions.
Lucia, I think your slides are not working properly. You maybe have to put a presentation mode.
I'm already in the presentation mode.
We don't see that.
Can you see it now?
We see the slides, but no in the presentation mode. So you have to move the slides from the side, maybe.
Now?
No. But I mean, we can see your slides, but not in the presentation mode. So if you want to change them, you can change them from the sidebar.
Well, I really don't know how to do that. Okay, maybe. I don't know how to do that, sorry.
Okay, never mind. Whenever you want to change your slide, just change it from the panel in the left. Because with the arrows doesn't work. So
like that?
Yes, like that. Perfect.
Okay.
Now, yeah, go on.
Okay, sorry.
It's okay.
So having a good electrolyte force field that faithfully reproduces the ion-ion and water-ion interactions is also fundamental when simulating proteins or nucleic acids, as their conformation depends upon the steric, electrostatic, hydrophobic, and hydrophilic interaction with the medium. It is also important for simulating anti-freezing proteins, which have very important applications in cryogenics and also in the development of new materials that are resistant to to extremely cold temperatures. Okay, so regarding the electrolyte models, we have unit charge models and scale charge models, and the first ones have been very commonly used, but they provide low solubility values, yielding anomalous crystallization. And one would expect that they at least would provide a good description of the solid state, but they overestimate the melting temperature by 20%. And so in this work, we have used the Madrid 2019 model that uses scale charges, and this scaling of the charges accounts for certain transfer of charge between the ions and the water surrounding them. And here is a list of properties that this model has already improved with respect to the description of unit charge models. So the question in this work was, will it be possible for the Madrid 2019 model to reproduce the maximum of density and its temperature for the salt solutions? And here I saw the results, and I want to clarify that it is very difficult to do experiments in the supercooled region of water because there are certain nucleation phenomenon that often makes the sample freeze before being able to measure it. And so here we have the results from Jacobo Troncoso at the University of Vigo, who has recently been able to measure electrolyte solutions at low temperatures. And the squares are the maximum of density, the temperature of the maximum. And the crosses are other experimental results at ambient temperature from La Liberté. And as we can see, the overall description of the Madrid 2019, which is represented by solid lines, is in very good agreement with the experimental data. And more importantly, the description of the density is not worsened at low temperatures with respect to ambient temperatures, as most of the models using unit charges do. Finally, we have also done molecular dynamics using the Young-Chipman model, which is one of the most used models with unit charges. And as we can see, it overestimates the density of the solution and underestimates a little bit the temperature of this maximum, whereas the Madrid 2019 model is able to reproduce quantitatively both the maximum and the temperature at which it happens. So the conclusions are that the Madrid 2019 model is able to reproduce with quantitative precision the maximum in density as well as the temperature at which it appears, at least 4 chloride solutions at 1 molal. So we can add these 2 properties to the properties that the model improves. And that is all. Thank you.
Okay, thank you, Lucia, for your nice talk. And now we do have time for some questions. I think we are a little late, but we have time for a discussion. So if you have questions, I encourage you to write them down on YouTube.
I have a question for Cynthia.
Okay.
How much time does it typically take for you to reach the equilibrium from the initial temperature?
Cynthia, are you around? Oh.
I mean, you mean computational times or like in real life times of calculation?
Computational times.
Yeah, so computationally, usually it takes maybe 2,000 nanoseconds or so. So usually around 2,000 nanoseconds or even more. In some cases, so it's an stochastic phenomenon. So that means we don't know when it's going to take place. Sometimes it takes longer, sometimes less. So it also depends on that, but there is another factor, that is how close do we start from the equilibrium. So depending on the initial configuration, this also changes. If the initial configuration is almost at equilibrium, it will take nothing for it to achieve it. But if it's quite far, it may take longer. We did not start that close. So yeah, 2,000 nanoseconds.
Okay, thank you.
You're welcome.
Great, great. I have a little question to Cynthia as well. How do you measure the solution concentration?
Okay, so for this question, I'm going to to try to share the.
Oh, okay, Lucia. So you have great.
Okay. So, okay, let's see if. It's in Spanish, but basically what we did is use the G density option of GROMACS. With that, we can take the density along the boxes for water and the ions as well. And then we identify where is the ice and where is the liquid. in this case, it's in between the two green lines. And for that, we get the average of the density of the ions and the average for the water density. And we divide it how it's shown up, and also dividing it for the molar mass of ions to obtain more per kilogram of solution. And then we can check over time how it evolves and average these blocks when we think equilibrium has been reached. I don't know if this solves the question.
Yes, that's perfect. Thank you so much. Since we are a little late, we're finishing the discussion to follow with the second block, which is magnetic materials. And our first speaker is Guiomar Delgado Soria. So if you're around.
Hello, yes.
Hello.
Okay, I'm going to share my computer. One minute. Okay, hello to everyone. My name is Guiomar Delgado Soria, and I'm going to introduce my work that is called Magnetic Behavior of Cobalt Films on Strontium Sapphiride. First of all, we need to explain the motivation that we are looking for an alternative for the actual permanent magnets, and a good material for this is strontium sapphiride. Why? Because there are a lot of promising magnetic properties like high magnetocrystalline anisotropy and elevate Curie temperature, good wear resistance, and the most important, that we have a high coercitive field. But there are an issue that the saturation of magnetization is relative moderate. So it's well known in the literature that when you combine with a soft material, a metal layer, in our case cobalt, you can improve the magnetization without a significant coercitive cost. But for this combination of the layers, we need to have a magnetic coupling between them. And for this, we did a different experiment. First of all, we did a magnetic coupling between a platelet of strontium sapphiride with a cobalt overlayer. The platelet sapphiride we characterize by means of X-ray techniques in alpha synchrotron in B-line CIRSE. And in this image, we can see the SAX XMCD spectrum that reveals the electronic transition in iron LH. It's important that the information of this graphic is the characterization in iron oxide state and the crystallography of iron, as well that we can determine the magnetic domains in the platelet. The change in the contrast, black to white, correspond to the different energy of photons that reveal that we have a magnetic, only magnetic domain, and this is out of plane. Then we growth a cobalt overlayer by a molecular beam epitaxy. And in this case, I think that is really interesting, this slide, because we can achieve the different magnetic domains in the cobalt layer and in the iron. On top correspond to cobalt layer. We take different XMCD images that correspond at big range contrast magnetic in this layer, in the case of the cobalt. And if you see the images in comparison 0 to 120, we can see that the domains change a little bit the contrast. When appear the region white, you can see black. So that is mean that we have a magnetization within the plane. But for example, in the strontium sapphiride platelet, we see the iron LH. The images in all the cases of the angles, we have all the time black domain that correspond to out of plane magnetic domain. So there are no correlation between the magnetic domains between both layers. Our results are interpreted like the shape anisotropy of the cobalt layer, dominating over the strontium saccharide out-of-plane magnetization. So the first goal in our project is looking for the magnetic coupling between both layers. So we did another experiment, and in this case, we grow a thin film of strontium saccharide within the magnetization in plane, and who is the behavior of cobalt layer with this interaction. First, we grow the thin film strontium saccharide by magnetron sputtering, and we determine his structure and magnetization by different spectroscopy and diffraction techniques. We emphasize in our work with Mossbauer spectroscopy, which provides information of the iron sites in the crystallography structure, as well as the magnetization in the thin film. We determined that the layer, the sample with the layer growth at the highest power deposition, as well the highest thickness, has the magnetization within the plane. This is in good agreement with the result in X-ray diffraction, which determine the EC axis is within the plane. Knowing that the EC axis in the strontium saccharide is in C plane is in good agreement that I said that we have magnetization within the plane. Then we grow a cobalt.
Guillermo, you have to finish already.
Perfect, because this is the last slide, sorry.
Great.
Yes, the last slide corresponds to the vector magnetization in each layer. We grow cobalt on top of the strontium saccharide thin film, and in this case, we have a region red and green. This corresponds to uniaxial anisotropy in the case in strontium saccharide. The beautiful of this is the cobalt layer has the same color. So we have the same EC axis in this part. But if you overlapping both images, like we can see this, the regions, the lines correspond to different magnetic domains and doesn't correlate. So we think that we don't have a magnetic coupling between both layers, but yes, a structural coupling. And thank you for your attention. I'm sorry so much.
Thank you so much. So our next speaker is Eduardo García Martín.
Now, can you hear me? Yes,
perfect.
All right. Okay, perfect. I'll share my screen. Second. Here. Okay, what you see the presentation? I think yes, but I'm not sure. It's all already okay?
No.
No.
We don't see the presentation mode. I mean, yes, you have to click that. Perfect.
Okay,
now,
perfect.
Yes, now.
Okay. Hello, everyone. My name is Eduardo Garcia. I am doing my PhD in the SURF-MOS group in the Institute Rocasolano and the Applied Physics department in the Autónoma of Madrid. I want to talk about my work, growth and characterization of thin films of lithium fluoride. Okay, and over the last 100 years, technology has undergone an enormous evolution, focusing in two aspects: the reduction of the size of these components and the speed of response of processor. This evolution was predicted, given rise that become known as Moore's law. This law establishment that every 2 years, the number of the transistor per unit area will be double every year. This predicted that 2027 as transistor gate will be so small that it will contain only 10 electrons. Having too few electrons creates two main problems: overheating and fails due to quantum interactions. For this reason, multiple branches of science based on nanotechnology are currently being studied that allow us to solve it or minimize these problems. In our case, we are focused on spintronics. Our objective is to synthesize a material that generates heat in use, little heat in use, low dissipation, and on the other hand, allow us to handle speed as fast or faster than currently materials, ultra fast. There are many possibilities, but now we've chosen a type of material that nanoionic materials. These are a material that allow through the application of the electric field to control migration of ions, how they move through the lattice, and to vary the properties physical like electrical or magnetical. We have chosen a lithium ferrite. The material has very good qualities. Has two structures, alpha, which the lithium is ordained and ordered, beta, which it's not. Their behavior is obviously different, but both cases are very interesting properties. Alpha beta is less well studied. This material has already been grown. The most used technique for that was PLD and pulse laser deposition and CVD chemical vapor deposition. These are interesting and useful techniques, but we decide to innovate. We are going to use system dips, double ion beam sputtering. These techniques allow grow very homogeneous sample and high crystalline quality. We explored several parameters like target, like partial pressure of oxygen, substrate, temperature, and energy and current density of sputtering and assistance. Regarding the characterization of the sample, we use structural characterization techniques like Raman spectroscopy, X-ray diffraction, nuclear reaction analysis, Rutherford backscattering, and for the properties electrical magnetic, we use MOG, magnetical optical Kerr effect, and 4 wires for property like resistivity. The early
excuse me, you have 30 seconds left.
Yes, finished. At early we had problem with lithium concentration, but we are on the way. We explored a new stoichiometry in the target, which allowed to have enough lithium in the material to develop the structure we want. Is like conclusion, we have obtained different phase on LFO, and we can control amount of lithium and the structure depending on the parameter like temperature, pressure, oxygen, substrate, and energy. And I won't finish, and thank you of everyone to attention.
Thank you, Eduardo. So our next speaker is Laura Álvaro Gómez. So Eduardo, you have to stop sharing.
Yes, I can. Yes. Yes.
Great,
yes.
Great. Thank you. So Laura, we cannot hear you.
Now?
Yes, now we can.
Oh, yes. So I remove my. Okay, so share my screen. Okay. Okay.
Perfect.
Perfect. So good morning, everyone. My name is Laura Álvaro Gómez. and I am a second-year PhD student at University Complutense of Madrid in the nanoscience in Madrid as well, and then in joint collaboration with Grenoble in France. So during this brief talk, I'm going to be talking about the micromagnetics of chemical barriers inserted within permalloy cylindrical nanowires, with the objective of controlling domain wall motion. So just as a brief introduction is the motivation of moving from two-dimensional to three-dimensional nanomagnetism. Because what happens is that in two-dimensional nanomagnetism, we have just magnetic textures that are confined into the two-dimensional system. For this reason, if we move to a third dimension, first we're going to be able to pattern new magnetic nanostructures, and then with that, we are going to be able to tune new magnetic properties. With that, we are going to be one step closer to the desired, well-known concept of racetrack memory. So in this memory, the data would be stored into the magnetic domains of a nanowire, and these magnetic domains may be moved by applying spin polarized currents. So far, we have experimental evidence that domain wall motion in cylindrical nanowires can achieve ultra-fast velocities. So then moving to cylindrical nanowires, we may have two types of magnetic domain walls. The first one is the transverse Borg-Test wall, and it has the same topology as nanostrips, which is transversal magnetization. And the second one is the block point wall. That is a domain wall that is unique in this system, and due to its topology, it can achieve ultra-fast velocities. But what happens is that it is a bit meaningless to have ultra-fast velocities of domain walls if we cannot control their movement. For this reason, we propose a system that is based on a nanowire with effective spinning sites to stop the magnetic domain walls. In this case, we're using a nanowire made of permalloy with chemical barriers of the inverted composition. So these nanowires are grown by single path electrodeposition using voltage pulses. Once we have synthesized these nanowires, it's time to do the three-dimensional magnetic characterization. So to do that, we make use of X-ray facilities in the synchrotron. In this case, I'm going to show images of PIM, STXN, and picography, where we can have access to the volume information of the magnetization. So just to summarize all these images, what we observe is that at the chemical barriers, we have a bipolar black and white contrast. That means that the magnetization is curling around the wire axis at the chemical barriers, whereas in the rest of the wire, the magnetization is axial along the wire axis. So this is the first conclusion. These nanowires have curling magnetization at the chemical barriers, and then we need to access this curling to fully understand it. So the first approach we do is to apply axial magnetic field along the nanowire axis to reduce this curling. So we can see that the magnetic contrast decreases as we increase the magnetic field. And then by using a code that we have developed in the lab, we can have access to the curling angle at the surface of the nanowire and see how it decreases as we increase this axial magnetic field. Then the second approach that we took is by applying nanosecond current pulses. So to do that, first we contact electrically in the clean room the nanowires by using a process based on laser lithography. And then we are going to apply current pulses. So when we start with this initial state, we see that the curling of the chemical barrier has a defined circulation, which is in this sense, and is white and black. And what happens is that when we apply a current pulse, the circulation of the curling is reversed. So if here it was white and black, now it's black and white. And it is reversed to the circulation of the Oyster field that was applied. So this is a very important conclusion. That means that the Oyster field drives the switching of these chemical barriers. And this is very key to understand because then the Oyster field has a high impact on domain wall motion. And just to conclude, I'm going to share with you some theoretical models that we are doing to understand better this curling and then to understand the interaction with domain walls. So the first one is in micromagnetic simulations that we observe that the curling has a limit because as we increase the size of the modulation, this curling increases, but there is a point that occurs at 60 nanometers where it reaches a plateau. So then we see that there is a limit in the curling. And then to understand it better, we perform an analytical model where we see that the curling occurs to have volume charges that can screen the surface charges at the chemical barriers. So then when we model a complete screen of volume and surface charges, we observe again that maximum coupling is not possible due to the cost in exchange energy. So this is a bit of what I wanted to show today. I would be very happy to answer any questions if there are any. Thank you very much.
Thank you, Laura. So our next speaker is Claudia Fernández-González.
Can you see my screen?
Yes. But
No.
not now. Yes, that's perfect.
Okay, so hello everyone. My name is Claudia and I'm going to speak about the scaling up the electrodeposition of nanowires for application in permanent magnets. As all of you have already known, permanent magnets are one of the components of a wide range of technological applications. Most of them contains rare earths, and these materials are well suitable for this application because they present a high energy product, which in other words is the parameter that determines the force of a magnet. But these rare earths present several problems related with their extraction. They are extracted in very few locations around the world. This extraction is environmentally harmful, and also it's related with production limitations, which increase the prices of these materials. So nowadays, one of the challenges in the field of the permanent magnets is to increase this energy product of other hard materials like ferrites in order to fill the energy gap that you can see here, in this graphic. So one alternative is to combine hard magnetic materials with a soft magnetic phase in order to increase this energy product. In this work, we propose the synthesis of a composite material of strontium ferrite powder and iron cobalt nanowires to synthesize a composite bonding magnet. We will focus our work in the development of a synthesis process for the nanowires production. And for that, we have chosen template-assisted electrodeposition because we can control the diameter, the length, and the composition of the nanowires in an easy way. So if we think in the production of a bonding magnet of 300 milligrams, we will need to produce 90 milligrams of nanowires. And in a common laboratory process, using polycarbonate template, we were able to obtain 0.01 milligrams of nanowires. So we need 4 orders of magnitude more to provide the amount of nanowires to produce a bonding magnet. So this work has 3 main objectives: to increase the mass of the produced nanowires, to reduce the synthesis time, and also to reduce the cost of the process. So first of all, to increase the mass of the produced nanowires, we decided to change from polycarbonate templates to aluminum. This allowed us to increase the pore density of the templates and also their thickness. And we were able to increase in 2 orders of magnitude the quantity of nanowires that we were able to produce. To reduce the synthesis time, we have focused this step in reducing the synthesis of the template because it was the most time-limiting process. So we modified some parameters of a common 2-step aluminum anodization, and we reached the production of 5 templates per day. And in order to reduce the costs, we changed the raw materials. We changed high purity aluminum for recycled aluminum, and also we changed the material of the counter electrode in all the electrochemical processes. And with that, we decreased in 3 orders of magnitude the cost of the production of the nanowires. So once we were able to provide the amount of nanowires needed for the synthesis of the composite bonding magnets, our colleagues Jesús Guzmán and Adrián Quesada from Instituto de Cerámica y Vidrio synthesized the bonding magnet with improved energy product and also magnetic remanence. So as main conclusion, the scale-up process of the nanowires production presented here opens the possibility to integrate these nanowires in industrial applications. And thank you so much for your attention.
Thank you, Claudia. Thank you to all the speakers. we do have questions in YouTube. So first, to Laura. Juan de la Figuera wants to know how delicate are the nanowires and how many did you destroy to get the data?
Okay, so thank you, Juan, for your question. It's a very interesting question because it's true. When you contact electrically the nanowires, there is a lot of risks for these charges, even from our hands, and they are burned. So after the clean room process, I would say just 30 or 40% of them are alive. And then after manipulation in experiments, I would say like maybe just 10% survive. And then once we image them in the synchrotron, sometimes they were burned in the modulations. So to improve that, now we have performed a coating of the nanowires to don't get explode at the chemical barriers. So quite a lot of them get burned.
Thank you. So Juan, if you have any further concern, please feel free to write in the YouTube chat. We have another question now, this time to Eduardo. In the future, you would like to study movement of lithium ions. How can this be done experimentally?
Yes, we won't do it in the future, but we know that the batteries, ion battery, use, for example, technical electrotechnical techniques for composition and know what is the movement in the lattice. But we are working in progress to know what is the best technique that allow the movement of ions. We are now concentrate focus in have the structure alpha well done with low defects, lithium, and nice stoichiometrical composition. Sorry, but I can't respond the respect the technique.
That's okay. Thank you so much, Eduardo. So if there are no more questions, and since we are a little late, maybe we can stop the discussion here. I don't know what Rebecca thinks about it. There is some noise.
Oh, is there?
Yes, a weird noise. Now it's okay.
Oh, yeah, I can see some messages. I can see some messages. Hang on a second. Everyone is silenced and there's no... Is it okay now, Luciana?
No, there is some noise when you speak.
With the microphone.
Like a really loud noise.
I will stop my microphone because maybe that's the reason. I don't know. I will, and you'll let me know.
The noise is gone, so I think it's your microphone.
So it's back there?
Yes, it's back.
Oof. And is it impossible to... Okay, what? Okay.
Okay, I can go on if you want with the fourth session.
Yeah, let's do that. Let's go to the fourth session.
Yes, we have our chair, Miguel Mompean. So Miguel, if you would like to go on.
Thank you, Luciana.
You're welcome.
Hi, everybody. Good morning. Thank you for joining us in this beautiful symposium. I'm Miguel Mompean from the Protein Nuclear Magnetic Resonance Group, and I'm going to moderate this last session. So our first speaker is Antonio Joaquín Monera. Antonio, please, whenever you're ready.
Sí, un segundo. Vale, ¿se ve todo bien? Vale. Bueno, buenos días, soy Antonio Monera y estoy haciendo un doctorado en el área de genética de la Universidad de Murcia. En mi grupo, Bueno, voy a hablaros sobre cómo hemos descubierto una enzima muy buscada en humanos trabajando con una bacteria del suelo. Esta bacteria, llamada Myxococcus santus, es capaz de percibir la luz y sintetizar carotenos para defenderse del estrés fotooxidativo, adquiriendo esta coloración roja tan llamativa. Hay una proteína llamada CARF que es esencial para que Myxococcus santus sienta la luz. Durante años pensamos que CARF podía detectar de alguna forma el oxígeno singlete generado en la luz y desencadenar la carotenogénesis. Sin embargo, voy a comentar que esto realmente no sucede así. CARF tiene un patrón de histidinas muy inusual que recuerda al descrito en desaturasas de ácidos grasos. CARF tiene proteínas homólogas en eucariotas, tanto animales como plantas, y unas pocas bacterias. Curiosamente, el CARF de Myxococcus santus se parece más a los homólogos de animales que a los de plantas. De todas estas proteínas homólogas a CARF, únicamente se conoce la función de la proteína AT3 de la planta Arabidopsis thaliana, que es precisamente una desaturasa de ácidos grasos. Teniendo esto en cuenta, nos preguntamos si CARF es una desaturasa de ácidos grasos. Para ello analizamos la composición de lípidos en una estirpe silvestre y una estirpe que carece de CARF. Y lo que encontramos fue la presencia de un pico en el wild type que desaparece por completo en el delta CARF. ¿Y con qué compuesto se corresponde este lípido? Pues con un plasmalógeno. Los plasmalógenos son un tipo muy particular de lípidos que son abundantes en animales, pero ausentes en bacterias aerobias, a excepción de las mixobacterias. Estos plasmalógenos se caracterizan por tener un ácido graso unido al glicerol no por un enlace éster, que es lo convencional, sino por un enlace viniléter, es decir, un enlace éter muy próximo a un doble enlace. En mamíferos, los plasmalógenos están implicados en procesos de señalización, diferenciación celular y fusión de membranas, y además son potentes antioxidantes. Hay una desaturasa llamada PDS1 que introduce el doble enlace característico de los plasmalógenos. Sin embargo, la identidad de esta enzima ha sido un misterio durante décadas. Sabiendo que CARF está implicada en la formación de los plasmalógenos en Myxococcus santus, nos preguntamos si CARF podría ser en realidad esta desaturasa tan buscada. Por ello, analizamos la presencia del plasmalógeno y su precursor en una estirpe silvestre y una estirpe que no tiene CARF. Y lo que encontramos fue que únicamente la estirpe silvestre tiene plasmalógenos, mientras que el precursor se acumula en la estirpe que no tiene CARF. Por tanto, en base a estos resultados, concluimos que CARF es la desaturasa que genera los plasmalógenos en Myxococcus santus a partir del precursor con enlace éter. A continuación, nos preguntamos si los homólogos eucarióticos son capaces de suplir la falta de la proteína bacteriana en Myxococcus santus. Al realizar este experimento, comprobamos que únicamente los homólogos de animales, llamados Timen 189, son capaces de generar plasmalógenos, al igual que CARF, y por tanto restablecer la respuesta a la luz en nuestra bacteria. En base a estos resultados, concluimos que CARF y sus homólogos de animales son la desaturasa que genera los plasmalógenos, mientras que los homólogos de plantas muestran una actividad desaturasa totalmente distinta, como es el caso del homólogo FAT4 de Arabidopsis thaliana. Por tanto, en conclusión, gracias a nuestro trabajo sobre la respuesta a la luz en una bacteria muy humana, hemos descifrado la identidad de una enzima muy buscada durante décadas. Ahora que sabemos quién es esta enzima, podemos eliminarla y analizar las funciones de los plasmalógenos y su papel en enfermedades. Para finalizar, quiero comentar que estamos intentando entender cómo las mixobacterias utilizan los plasmalógenos para ver la luz. Este es un nuevo mecanismo de señalización que proponemos, que aún estamos tratando de demostrar, en el que nosotros pensamos que los plasmalógenos se rompen por el oxígeno singlete generado en la luz. Muchas gracias por vuestra atención y agradecer la financiación recibida.
Thank you, Antonio. thanks for keeping this in time and for the beautiful talk. So our next speaker is Ricardo Pérez.
Sí,
Ricardo,
comparto la pantalla.
the screen is yours.
¿Se ve bien?
Yes.
Vale. En esta charla voy a hablar de la enorme plasticidad que hay en el modo de acción de una nueva clase de fotorreceptores basados en la vitamina B12. Como ha comentado mi compañero Antonio, Mysococcus santus, para protegerse de los efectos oxidativos, fotooxidativos de la luz, produce carotenos. Y fruto del estudio de cómo se regula esta síntesis de carotenos, se ha visto que Mysococcus santus utiliza dos vías. Una vía independiente de B12, que es la que implica GARF y la que ha explicado Antonio, que es bastante compleja, y otra ruta más directa en la cual actúa el fotorreceptor CARH, que está basada en la vitamina B12. La vitamina B12 es esta molécula que vemos aquí y tiene un interesante enlace cobalto-carbono que es fotolábil. Por lo tanto, la luz lo que hace es romper este enlace cobalto-carbono y romper la molécula. Por lo tanto, en la luz, CARH ya no va a estar unida a adenosilcobalamina, que es la forma de vitamina B12 que une CARH, y por lo tanto ya no puede reprimir la expresión de los genes carotenogénicos, por lo que las células producen carotenos y se vuelven rojas. Nuestros análisis han demostrado que CARH está ampliamente distribuido en bacterias y nosotros hemos estudiado diferentes homólogos de CARH de estas bacterias. Por ejemplo, hemos estudiado el homólogo de CARH de Mysococcus santus in vivo, porque in vitro no se pudo porque la proteína fue insoluble. Pero se vio que Thermus thermophilus, una bacteria termofílica, tenía un homólogo de CARH y que sí que fue soluble. Y fruto del estudio de este homólogo de CARH pudimos analizar in vitro la proteína y hacer análisis estructural y de la fotoquímica de la proteína, del fotorreceptor. Y en mi tesis estoy centrándome en el estudio del homólogo de CARH de Cystobacter fuscus, una bacteria de la familia de Mysococcus santus, y de Bacillus megatherium. Estas proteínas sí que fueron solubles y se han analizado in vivo e in vitro. CARH es una proteína con dos claros dominios, un dominio N-terminal de unión al ADN, separado por un linker de un dominio de unión a B12. En la estructura se ha visto que hay, en cada uno de los dominios hay aminoácidos importantes para la función de esta proteína. Yo me voy a centrar en la enorme plasticidad que hemos visto en los diferentes homólogos que se han caracterizado de CARH. En Thermus thermophilus, la forma APO, es decir, no unida a la vitamina B12, es un monómero, mientras que en el caso de Bacillus megatherium es un glóbulo fundido, un tetrámero. Y en Cystobacter fuscus es un dímero. Esta forma APO, cuando une en oscuridad adenosilcobalamina, en el caso de Thermus thermophilus tetrameriza, ¿vale? En el caso de Bacillus megatherium se forma una estructura rígida y activa, y en el caso de Cystobacter fuscus el dímero sigue siendo dímero, pero ahora activo. Y estas proteínas con la adenosilcobalamina unida son capaces de unirse al ADN para reprimir la expresión de los genes que llevan aguas abajo. En este caso, de genes carotenogénicos. En el caso de Thermus thermophilus, utiliza 3 de sus 4 dominios de unión a DNA para unirse al ADN y se une utilizando 3 repeticiones directas. El operador de las proteínas es parecido, pero en el caso de Bacillus megatherium utiliza los 4 dominios de unión al ADN para unirse a su operador, y en el caso de Cystobacter fuscus se une 2 dímeros a su operador para reprimir la expresión de los genes. La luz provoca la fotólisis de la adenosilcobalamina y en el caso de Thermus thermophilus se libera del DNA porque se rompe la adenosilcobalamina y el tetrámero produce ahora monómeros. En el caso de Bacillus megatherium el tetrámero da lugar a dímeros y en el caso de Cystobacter fuscus el dímero da lugar a monómeros. Toda esta plasticidad en el modo de acción de los homólogos de CARH es importante porque se ha visto que estos fotorreceptores tienen un montón de aplicaciones optogenéticas. Otros grupos de investigación han aprovechado la investigación básica hecha en nuestro laboratorio para controlar la expresión de genes. Nosotros lo hicimos en bacterias, pero otros lo han hecho en mamíferos y plantas.
Ricardo, could you please finish?
Sí, es lo último ya. Utilizando la capacidad de oligomerización de CAR-H, lo que han hecho es controlar diferentes rutas, es decir, la interacción de proteínas y diferentes rutas de forma dependiente de luz y B12. Y también algo muy interesante que han hecho es generar hidrogeles que cuando están en oscuridad y con B12 encapsulan proteínas y células. Y entonces cuando le da la luz, de una forma controlada por luz, liberan estas proteínas y las células. Y hace, por ejemplo, 2 semanas aplicaron CAR-H para controlar la liberación de insulina en un ratón diabético. Este reloj monitorea la concentración de glucosa que hay en sangre y cuando ve que la glucosa es baja, da una luz verde y entonces ahora CAR-H no puede reprimir el gen de la insulina y se expresa. Muchas gracias.
Thank you, Ricardo, for this excellent talk. So now we can move to the next speaker, which is who is Miguel Daniel Mozo. Miguel, you there? Ricardo, can you please switch off your microphone?
He parado, ¿no?
Yeah, your microphone.
Yeah.
There's the silence
A
button.
ver. Okay, can you hear me?
Yes.
Okay, so awesome. Okay, so good morning, everyone. Thank you for the opportunity. And as Miguel just said, my name is Miguel Daniel. I'm a third-year PhD student in Armando Albert's lab in the Instituto de Química Física Rocasolano. And in the next couple of minutes, I'm going to be talking about our studies on a plant calmodulin like CML23, which turned out to be a calcium and the pH coupled sensor, which modulates plant response to stress. Now, before I start, let me just give you a very quick introduction into why we're interested in stress and abiotic stress in general. So basically, where population is rising, and this means that food production and food supply in general is very important, especially when we take into account the major threats we have, such as soil or water limitedness, right, or global warming. So this arises a question, right? A question arises, which is, how are we going to feed 10,000 million people by 2050, right? So the answer is we need to generate and engineer plants which are more resistant to stress, so we can obtain better yields from crops, right? Especially crops grown in abiotically stressful situations. So one of those abiotic situations I want to focus my talk on is saline stress and drought, right? Because sodium is a toxic ion for plants because it induces potassium starvation through competition, right? And potassium is a vital ion for plants. So plants have to activate rapid responses to avoid potassium accumulation and maintain potassium homeostasis. So just moving very quickly through this slide, you can see that whenever there is an increased sodium uptake in saline environments, some of the sodium is pumped out through the sauce roots, right? But if there is too much, it will travel at the plant and towards the leaves. And in the leaves, it will alter potassium homeostasis, right? So in the leaves, there is a protein called NAX7, which controls potassium import into the vacuole. You can see it in this figure right here. And it acidifies the cytosol while doing so. It's been reported that the protein called CML23 interacts with it and regulates its function, but we don't know basically anything on the underlying regulation, and we don't know how this mechanism is controlled. So this is where we come in. In order to understand better this regulation, we crystallized and resolved the structure of CML23 at the 3-angstrom resolution, and it's the structure you can see right here. So basically, CML23 displays the typical calmodulin-like structure, right, of a calcium sensor with 4 calciums, 1, 2, 3, and 4 in the 4 EF hands, and also the domain distribution results in 2 cavities, which are opposite to the EF hand pairs. So you can see a small cavity, right, right here, sorry, and a large cavity, which we think is the binding site for NHX7. So when you think about how CML23 regulates NHX7, we thought, well, this protein binds calcium and it binds NHX7. So there you go. It has to be, like, calcium has to be regulating this mechanism, right? So to check this, we characterized the calcium binding affinities of CML23. We performed ITC experiments to calculate calcium affinities, and we double-checked with nano-DSF and a lot of other techniques, and we got to the conclusion that CML23 is a calcium sensor, and it displays two sets of sites with a sensing affinity in the micromolar range, right? But when we continued with experiments, with binding experiments to a synthetic CAM-binding domain of NHX7 in presence or the absence of calcium, we saw no difference in binding, right? And we were like, there has to be some other mechanism that's regulating the interaction, right? So if we go back to this slide, you might remember this, just I showed it 2 slides ago. So basically, NHX7 is exporting protons into the cytosol. So we went like, wait, maybe pH has something to do with this, right? Is it maybe a pH sensor? And that's the next thing we checked. So we performed circular dichroism experiments, and you see that when we compared a basic resting pH 8.3 CML23 with acidic stress conditions CML23, there is a big shift in the alpha helical content, and we double checked again with fluorescence studies, and we saw a blue shift right here, which means the protein is more stable at pH 6.3. So there's a big conformational change in the protein when it comes to pH, right? And so we went back to our binding experiments. Well, this confirms it is a pH sensor, right? So we went back to our experiments and we performed peptide binding experiments. This is my last slide. So basically we saw that the binding to the synthetic CAM binding domain of NHX7, when it came to pH, was almost 1 order of magnitude higher at pH 6.3 compared to pH 8.3, right? So we got very happy with this. And this proves that CML23 binds NHX7 preferentially at pH 6.3. So just some final conclusions, take-home message, CML23 is a calcium sensor at physiological conditions. It's also a pH sensor under these conditions, and it binds to NHX7 preferentially under saline stress acidic conditions. So thank you very much for your attention, and if you've got any questions, just please don't hesitate to ask. Thank you very much.
Thank you, Miguel. Thank you for this beautiful talk. So let's see if we have some questions here in the YouTube chat. Not yet. So maybe I'm going to start asking Ricardo if he could tell us a bit more on these optogenetic applications that can be envisioned based on the conformational plasticity of the homologs of this protein.
Basically, they are using mostly the Thermus thermophilus CAR8. That is a monomer in its isoform, but it's a tetramer in its holoform. So when it's bind adenosylcobalamine, it became a tetramer. Okay. So in this way, it became from monomer to tetramer. That's light controlled. So then with light, it became again a monomer. So they can control, but like when doing chimeras with CAR8 binding, CAR8 adenosylcobalamine binding domain, doing chimeras with proteins that want to oligomerize in a B12 dependent manner, they can play, and they have done hydrogels by fusion spike catcher domains. Others, they can make chimeras with a lot of proteins and control their function in a light and B12 manner.
Okay, but so you use light to dissociate this tetramer into monomers.
Yeah.
And is there any homolog that can do it the other way around? Because that would be more convenient, for example, to drive the formation of condensates or protein droplets. So do you know any homolog?
Maybe another CAR8 homolog that we haven't characterized do this. That's why it's necessary more basic research in studying those homologs, but not yet. We have seen that carriage of Bacillus megaterium is a tetramer that becomes a native tetramer that can bind to the DNA, but then became a dimer, and then Cystobacter fuscus is another kind of tetramerization or oligomerization. So maybe we find another, but not yet.
Okay, thank you. So do we have any more questions for our speakers here in the chat? We have a couple of questions here in YouTube. Miguel, this is from Douglas Lawrence to you. If you subtract the two CD spectra at the two different pH, do you get an alpha helical spectrum?
Yes. So the quick answer is yes. The longer answer is we haven't actually done that, but what we have done is we have compared the total alpha helical content at pH 8.3 and compared it to pH 6.3, and there is a big difference in alpha helical content. In fact, we are almost sure that the difference in alpha helical content is related to the C terminal of the protein, which is slightly disordered. It's forming an alpha helix, but it's not quite there. So we believe there are some contacts which can be created or destroyed according to the pH the protein is in, and this would make it this would make it helical or destroy this alpha helical C terminal domain. So we believe that is what's triggering the difference between the two pHs.
Okay, thank you. So the next question is also for you. It's from Maria Jose Sanchez. She's asking, what are the characteristics of the large groove to think that it is the binding site?
Yeah, so the groove is actually pretty, like the structure itself is similar to, as I said in my talk, it's similar to other CAM binding proteins, to other calcium binding proteins, but at the same time it's pretty distinct because of its C terminal, which is, it's unique basically. But still the groove is like typical when we've studied and compared it to other examples of calmodulin binding other peptides, and we have a model, or we think we have a pretty good model of how the peptide would bind it. There's usually a tryptophan residue in the peptide that would go inside the crevice, so it's a pretty hydrophobic crevice, and there are some interactions right there which would stabilize it. So that's why we're pretty sure that's the crevice where NHX7 binds. But I'm just going to link it to Juan Hermoso's question, which is right up in the chat. That's one of the things we still have to do. We have to make sure that the crevice, that the peptide is binding exactly to that point. And so we have some experiments designed to prove this and to show this.
So that's the question from Juan, which is what are the next steps in the continuation of the project? And there is also another question from Maria Jose. Given that the antiporter activation would remove potassium from the cytosol, are you thinking in ways to inhibit this interaction so that there is plenty of potassium in the cytosol?
Yes, let's see if I understood the question correctly. I think it's a really good question. So basically, there is some controversy on how the whole mechanism, how the whole inhibition of NHX7 works. But basically, we believe that whenever CML23 interacts with NHX7, something has to happen so that potassium stops going into the back wall and it stays as much as possible inside the cytosol, because sodium is toxic for plants and it will compete for potassium. So we have to raise potassium levels so that the competition is not affected, right? But we only want this to happen whenever there is sodium stressful conditions, abiotic stressful conditions for the plants. So we'd have to think thoroughly on how we inhibit this mechanism only whenever there is high sodium in the soil, right? So it's a very good question, but we're not into that exactly yet. We are still trying to understand the mechanism, and once we have that, then maybe we can move on and find a strategy to control this exactly the way we want it to.
Thank you. So if there are no more questions, we can move to the last part of this fourth session, which is on protein aggregation. And our first speaker is Jose Daniel Camino. Jose Daniel, whenever you're ready.
Hi, you can listen me? Good?
Yes.
Hi, I am Jose Daniel Camino. I work in Zaragoza in the Institute with Nunilo Cremades. I have just finished my PhD the last week, and I want to speak today about my job. I went to compare. I want to speak about clinically. My goal is characterization of primary nucleation of physiological relevant alpha-synuclein aggregation. Alpha-synuclein is intrinsically is a protein. Is a presynaptic intrinsically disordered neuron protein whose misfolding and aggregation in the form of amyloid fibril is the hallmark of different neurodegenerative disorders known as synucleinopathies, including Parkinson's disease. Amyloid nucleation has been observed to be initiated in vitro as a hydrophobic-hydrophilic interface by heterogeneous nucleation, generating parallel beta-sheet aggregates. Also, no such interface and be yet identified in vivo. With the aim of understand the different in vivo possible amyloid relevant alpha-synuclein mechanisms, we have characterized the amyloid pathway of alpha-synuclein under a variety of experimental conditions. And we have discovered that alpha-synuclein can assemble into amyloid aggregates by homogeneous nucleation, with alternate of active interface and with a preference for antiparallel beta-C arrangement. The typical aggregation procedure requires shaking or external interface to promote aggregation by heterogeneous nucleation, giving the formation of parallel beta-sheet aggregate, amyloid aggregate. We improve the kinetic reproducibility of this process using co-solvent without shaking to promote amyloid aggregation of alpha-synuclein. We could see that the concentration of co-solvent that is associated with a decrease of water activities, the kind of kinetic follow-up and the arrangement of the aggregate domain. We could distinguish between two kinetic regimens, one of long kinetic and the preference for parallel beta-sheet arrangement, and other that show short kinetic and preference for antiparallel beta-sheet arrangement. To study the primary nucleation of both kinds of regimen, we remove the hydrophobic hydrophilic interface with a hydrophobic cap, probing that aggregate goes through heterogeneous nucleation, because when we remove the interface, the aggregation is avoided. This occurs with long kinetic regimen. We work with the short kinetic regimen. We can see that the protein don't interact with the interface, because we can study the tension superficial of this interface with tensiometer. And for reinforce this result, we use custom setup because kinetic is so fast that require another protocol different to the hydrophobic cap for avoid the interface hydrophilic. With a microfluid made of hydrophobic material, we achieve aggregation, proving that the short kinetic regime go through homogeneous nucleation. This is very important because in vivo we can find a interface to mimic the interface that we work in vitro. And as conclusion, the result show us that homogeneous nucleation that believe high hydrated condition is so extremely slow. Is so extremely slow. It's not visible the aggregation. Under condition such hydration with under condition with such hydration, hydrophobic surface cold catalyzed nucleation through nucleation kinetic are still slow, but the nucleation can be catalyzed by reducing activity water and thus the hydration of the protein. Nucleation will be favored, in particular the heterogeneous nucleation under condition of moderate hydration. Under condition of reduced hydration, homogeneous nucleation is very favored, resulting in a fast kinetic aggregation and leading to antiparallel PTC arrangement. And I want to thank to my group and my collaborator.
Thanks a lot for this beautiful talk. So our next and final speaker of this last session of the symposium is Pablo Gracia. Pablo, when you're ready.
Okay, so can you hear me now?
Yes.
Is my screen sharing at all?
Not yet.
Okay, there we go. Cool. Is it good now?
Perfect.
Okay, I'm not sure if I'm honored or scared to be the last speaker here. So I'll present you very briefly the basics of the work I'm doing in my PhD thesis, also in the lab of Nunilo Cremades. So I'll spare you some of the details on amyloid aggregation and neurodegeneration because Pepe, Jose Daniel Camino has already presented that. So we work, our focus regarding biology is the amyloid aggregation of some proteins which play a major role on neurodegeneration. In this process of protein misfolding and amyloid aggregation, we encounter some challenges, mostly the heterogeneity of the structures and populations that appear along the reaction axis, as it is illustrated in this graph. And also recently, among these aggregation pathways, a new one has arose, which is called liquid phase separation, which is the self-interaction and further separation from the solvent of macromolecules upon certain conditions. So in order to tackle the aggregation property of some of these proteins, we are also collaborating on studying potential molecules that could bind the intermediate species or the mature species of this amyloid and abolish their toxicity, therefore. So we focus on both the drug interaction and the liquid phase separation of these systems. For the first of those, the interaction of small molecules with the amyloid particles, we encounter one main challenge, which is the heterogeneity of these interactions. So for that, we decided to use a single particle approach. We use single particle fluorescence spectroscopy, and we do that because I really like this quote of Stephen Chu. On average, humans have only one mammary gland and one testicle. So this, translated into the protein world, would mean that basically when we use conventional averaging techniques and we look at a sample, we see only the average signal, so the ensemble signal of all the proteins. But we want to look at one protein at a time, so to look at them individually to discern between populations and different behaviors. In order to do that, we use a modified commercial confocal microscope, which has a special sensitivity and excitation equipment, and also a very sophisticated time-correlated photon counting unit. I will say nothing else about this, but I'm very happy to discuss it with you later if somebody's more into the technique. So basically, we use a very dilute sample, and every time a particle passes through the laser beam, we detect their individual signals separately, as it is depicted in the graph. So for example, red could be the interacting molecule here, so a potential drug molecule, and green could be our aggregates of interest, right? So by measuring the intensity over time, together with the arrival time of each photon, we can then run a multiparametric analysis. I've depicted here in purple the techniques that I will share the data from with you in the next slides. So we applied this in collaboration with a Barcelona lab for the interaction of a small molecule with the neurotoxic forms of alpha-synuclein. And on the left side of the graph, you can see the individual peaks that correspond to the aggregates and the red ones which correspond to the small molecule. So by correlating the intensities over time of the two signals, we can have a single particle perspective of the binding. And we run this analysis separately by two different and independent methods. So we proved our bimodal analysis to be robust. As you can see in the graph on the right, we were able to establish the interaction affinity and stoichiometry of this complex by two independent analytical methods with almost the same results. So we were happy to publish this recently, and we are also trying to publish a methods chapter on this. And then, if you remember what I told you about liquid phase separation, we are currently also studying the LLPS and the liquid to solid transition of a different amyloid system. And among other techniques, we are using microscopy to look at the liquid droplets and the liquid particles. So instead of using intensity based microscopy, we are using lifetime based microscopy. So basically lifetime is the decay of the intensity of the fluorescence of the particle over time, and it is dependent on the on some of the properties of the solvent or the containers, such as pH, temperature, or crowding. So it can be used to add a new layer of information on top of the intensity layer. So here in gray is intensity, in colors it's lifetime. So this has enabled us, for example, to distinguish between two different environments during the liquid to solid transition of phase separated alpha-synuclein in a different amyloid system. And we are together with other techniques, we are also working on a manuscript on this. So basically, lifetime microscopy gives you a second layer of information that enables you to distinguish how your fluorescence probe is behaving in the same particle with different behaviors. So we could distinguish between young or mature droplets, or between liquid droplets and more solid-like ones that were leading to amyloid aggregation. I'm sorry if I went too fast, but I'm very happy to discuss everything you like here. I'm thanking my group, of course, and also our collaborators in the drug interaction project. And just let me mention that we are very happy to collaborate with every single one of you and everybody out there, especially using our microtime 200 single particle equipment. Thank you very much.
Thank you, Pablo. Excellent talk. So now we have time to discuss with Jose Daniel and Pablo. I'm going to check if there are some questions here in the chat. Not yet. Does anyone has a question? Otherwise, I can start with a question to Jose Daniel.
Yes.
If I got you right, you were speaking about an anti-parallel conformation of alpha-synuclein amyloid fibril, right?
Yes.
Do you expect that conformation to be more stable than a parallel one? Because when you say anti-parallel, you're talking about the stacking of the individual beta strands along the fibrils, right?
Yes.
And so, do you know, or are you aware of any prior EM structures of the many that are available from alpha-synuclein that display this anti-parallel fold?
Sorry, if I understand good, synuclein, until now, synuclein by electronic microscopy and be studied only by parallel structure, are no antiparallel. Antiparallel structure are be detected only for oligomer of synuclein, but not for mature fibrils. We are studying this with two techniques, infrared with pyramid fluorescent, but until now are not studied with structural study from this kind of topology. Neither cryo-IM, neither RM or another.
Okay, thank you. So we have one question here in the chat from Rebecca. Rebecca has a question for Pablo.
Yes, thank you, Miguel. I was just curious, Pablo, thanks very much for your talk. I'm curious about the source that you use in your fluorescence lifetime imaging experiments.
You mean the laser source?
Mm-hmm, that's right.
So those are laser diode heads. So they are argon laser diode heads.
Okay, and do you need the pulses to be particularly short, or do you need some?
So the length of the pulses we adjusted depending on the lifetime of the fluorophore. So basically, when you excite the fluorophore, the fluorescent signal decays over time according to its lifetime. And because we use a dual color approach, so we are able to distinguish two different particles in our system, we need each of the lifetime decays not to interfere with each other. So we need the laser pulses to be separated enough so we allow one decay to finish before the other one starts.
Okay. Thank you very much.
Sure.
We have another question for Pablo in the YouTube chat. Pablo Miguel Garavis is asking, did your heat compounds avoid aggregation processes?
Yeah, so first of all, I would like to say that the compounds were developed and envisioned by Salvaventura's lab, as it was in my last slide. We were doing the experimental part of characterizing the interaction. And yes, they do inhibit amyloid aggregation even more strongly than one compound that was published recently, which is called C-nuclein D, which was also very promising in the field. So they block aggregation down to like a 10-15% of the wild type, of the uninhibited process. And particularly, we have found that it interacts with the early oligomeric forms of the protein, and it blocks the conformation of the protein in the oligomeric form. It arrests pretty much all the protein mass in the solution to these oligomers, but these oligomers turn out to be benign in this case, or their toxicity is reduced. And we have also seen that by the cell membrane binding and binding disruption experiments that they performed. So yeah, so in this case, strong binding to the toxic oligomers corresponds to a protective effect. And also let me tell you that it was very interesting to see how, of course I didn't have the time to explain this, but these inhibitors bind specifically the toxic species of synuclein, and it does not bind neither the physiologically active monomer nor the benign disordered oligomers. And we have also established the mechanism for this. which is through a combination of a hydrophobic patch binding and a cationic character of the inhibitors. So that was actually the main point of the article. Thanks for the question, by the way.
Thank you, Pablo. We have more questions from our audience, in YouTube or in Zoom. I'm seeing more questions. So, Pablo, I would like to ask you something also.
Sure.
In the first part of your talk, you showed this beautiful characterization of the compounds with the interaction with the alpha-synuclein using this microscopic technique where you follow the intensity over time. And you have two colors, right? So
yes.
Can you use that to detect mixed amyloids, where you have an amyloid formed by two different proteins?
Sorry, can you repeat your question, because I couldn't hear it properly, sorry.
Yeah, because you know that there are some amyloids that in the fibril you have two proteins.
Heteroamyloids, yeah, sure.
Protein.
Yeah.
And with the proteins that you have presented with it is quite easy to make these mixed amyloids.
Right.
And I think that this type of microscopy was used to visualize two different proteins within the same fiber. So is it the same method that you were? Not really, right? Can you explain?
Not really. So I guess what you're describing, it would be super resolution microscopy, which sometimes is also used for single particle tracking and so on. So here we don't have that spatial resolution. We have a very good very good time resolution. But so in the single particle spectroscopy experiments, what you see is the diffusion or co-diffusion of two different colors in time. Mm-hmm. And in the microscopy experiments, in the FLIM lifetime microscopy experiments, what we actually see, it's not in the presentation, but we are now working on a manuscript on this. We see that two different amyloid proteins co-aggregate in one of these liquid phase separated systems. But we cannot see it at a single fiber level at all. I mean, that would be super resolution microscopy. But yeah, I'm aware of the topic. It's very, I mean, I think you're also very well aware of heteroamyloid and so on. So it's a very interesting field too.
Okay, thank you.
Sure.
So let me check if there are.
So sorry, can I just clarify one thing? I mean, of course, with the single particle techniques, we can tell certainly whether one particle has both proteins, so whether one aggregate particle has both proteins. Mm-hmm. But it's not it has pretty much no structural resolution. That's what I meant. Mm-hmm. So you can colocalize it over time, you can measure their co-diffusion, and you can calculate the stoichiometry of the complexes. But for structural resolution, you would need to resort to either FRET or super resolution. Sorry for interrupting you.
I think they call that this technique where I'm referring to. They call that fluorescence polarization, where they have the fiber with the two colors, and then they count every time a fiber passed through the detector. Mm-hmm. They say the intensity of the two colors at the same time. That means that you have the two proteins in the fiber.
I mean, that is exactly what we do with small molecule binding to the alpha-synuclein amyloids. We can do exactly the same for amyloid interactions. What I was trying to say is that you're able to tell the stoichiometry of the complexes. You're able to tell whether both proteins are existing in the same aggregate, mm-hmm. but you have no structural resolution.
Oh yeah, that's great.
Okay, sorry, I got it wrong altogether.
That technique that you showed, because you showed for the small molecule with alpha-synuclein, if that could be used to scrutinize if the two proteins are in the same fiber. So
yes, you can do that. I mean, we're doing that with imaging, and we have also tried to do some in-solution spectroscopy for it. It has some more challenges, but you can certainly do that. Yeah.
Mm-hmm. Great, thank you.
Sure, welcome.
So if there are no more questions, and I think there aren't, I think, Rebecca, you would like to finish this session, right?
Yes.
I'm going to mute myself.
Thank you very much, Miguel. well, I guess my previous problem with the audio is solved. No, it's okay now, right? Okay. Well, thank you very much to everyone. We are at the end of this 7th edition of the Young Researchers Symposium. Well, I have to really thank the speakers. You've done a tremendous effort to condense all your work in these 5 minutes. You've done a great work. Now we know a lot more of what is going on in the Institute and in the associated units. Best of luck for those who are going to defend your thesis soon, and congratulations to those who've just done it. Okay, thanks also warmly to the chairs, Nuria, Inma, Luciana, Miguel. You've done a great job. You've been essential to make this work and to encourage this lively discussion. I also have to thank Esther Rebollar, who's been working with the YouTube emission and the YouTube chat. Thanks to all of you who are there at the other end of the YouTube emission and who've been following the symposium and participating and asking questions. We hope that we can meet in person next year. This format does have some advantages, and one of them is, of course, that one can participate from anywhere in the world. But of course, in some sense, we miss some more human contact. So let's hope that next year we can meet in our Salón de Actos. For those who've missed maybe some parts of the symposium, the video will be in the YouTube channel of the Institute. So thanks everyone, and see you next time, and best of luck with your careers. Bye-bye.